ഇതായിട്ട് നമ്മളല്ലല്ലോ
അവര് വലുതായിട്ട്
അവര് ഇടുന്നതാണ്.
റോസ് റോസ്
ലാപ്ടോപ്പിൽ
ഇടണോ
മിനിമൈസ് ചെയ്തത്
നോക്കേ ഗൂഗിളിൽ കയറി ഇത് ചെയ്യും പക്ഷെ ഓ ശരി ഇതുകൊണ്ടാണ് കട്ടായി പോകുന്നത്
നമ്മളെ മെയിൽ ഓൾറെഡി ഓപ്പൺ ആണല്ലോ.
Good morning, everyone. Welcome to the day two of our symposium. Definitely, you all had an amazing time yesterday, and we are thrilled to have you back to another day of insightful discussions and networking. So, appreciating everyone for joining us on a Sunday morning. So, thank you so much, everyone, for joining. So, today is also scheduled and packed with so much of exciting presentations, and definitely we will be having some insightful Q&A also. So before we kickstart day two symposium, we have some announcements that all our delegates who has registered it will get e-certificates on your email IDs. And top 20 young investigators can collect your participation certificates from the counter that is at the outside. That will be our registration desk. So make sure that the top 20 young investigators are collecting your certificates from the counter outside. And also, those who have registered it, you will get your e-certificates in your email IDs. And ladies and gentlemen, we will be kickstarting our day two within 10 to 15 minutes. Let's wait a few more minutes to all our people will be joining with us. Thank you. So, ladies and gentlemen, once again, welcome to day 2 of our symposium. And without further ado, let's dive into the scientific session 3, that is our first session in day 2. And the topic is role of gut microbiota and probiotics in cancer and metabolic disorders. And I would like to welcome the chairpersons. So put your hands together for Professor Chandrabhas Narayana and Dr. Sabu Thomas. I request both of you to welcome and introduce the keynote speakers.
Good morning, everyone. It is a lovely day, and I hope all of you enjoyed your deliberations yesterday, as well as the poster session. I don't know how many of you actually visited. Their posters are really excellent quality, and I hope all of you would interact with these young investigators. I hope today you're going to have another feast of talks, which will be of excellent quality. And I hope somebody can call the young investigators to come back. They're all standing next to the poster. I hope somebody can go and ask them to come and attend this thing. So welcome to this session. And I am co-hosting this with my colleague and current director of Center for Microbiome, Dr. Sabu Thomas. Unfortunately, I think the organizers put the wrong photo in there. This is the right Sabu Thomas I'm talking about. I just wanted to mention that. Okay. And I have the pleasure of introducing the next speaker. She is Dr. Adra Bhatt. She is currently the assistant professor at Department of Medicine, Division of Gastroenterology and Hepatology, School of Medicine, University of North Carolina in USA. Her lab is applying multidisciplinary techniques to study reciprocal host microbiome interactions, particularly as they relate to the host xenobiotic and endobiotic metabolism. It's a great pleasure to have you here, and I hope you are. This is your first visit to Trivandrum, and I'm hoping that you are having a wonderful time here. And we hope to have you many more times in Trivandrum. Welcome, and let's put our hands together to welcome Dr. Adra Bhatt. She also happens to be from Gujarat, right?
Yes, Gujaratis in the house. Let's get some, come on. Hey, good morning, everyone. I would be remiss if I didn't start with a really big thanks to Professor Ganguly, Dr. Hajela. Thank you so much for this gracious invitation and for your hospitality. I'm just delighted to be here. I'm so impressed by all of the science that's going on in microbiome research here in India, the country I call my home country. So I'm just delighted to be here and share my work with you. And I apologize at the very start. I'm going to try to run really fast so I can stick to the time limit. But I'm happy to talk to anyone after, like during any of the breaks or over email. My email is on the last slide. Please don't hesitate to contact me. And I apologize if I talk really fast. But so yesterday we had some amazing talks in which we learned about how bugs, like I like to call it, just, various bacteria, bugs are being used as drugs. We saw multiple examples of bugs being used as drugs for neuropsychiatric conditions, malnutrition, etc. And it covered the gamut from prebiotics, postbiotics, and symbiotics. I'm here to tell you about something very different. So instead of bugs as drugs, I'm going to talk to you today about drugging the bug and how that can be used to improve precision medicine. And by drugging the bug, I mean beyond using antibiotics, which we know are very harmful for our indigenous microbiota because they can eliminate them. We know that our gut microbiome is essential for so many aspects of homeostatic wellness, well-being, etc. One thing that we don't talk about very much is how the microbiota impacts drug response. And for the length of this talk, when I talk about drug response, I'm going to be talking about efficacy and toxicity. So there's a lot of emerging evidence, including some from my lab, that implicates gut bacteria in modulating the response to various medications. So we know that medications can change the microbiota by, altering the pH, the osmoticum, barrier integrity, nutrient availability for microbiota, as well as exerting bactericidal and bacteriostatic effects that are independent of the actual antibiotics. So we know that a very important class of medications and the antipsychotics are often associated with pretty strong bactericidal activity, which was not very well known until just the last decade or so. In work from my lab and others, we're finding that microbiota can, in reciprocal fashion, alter the efficacy of medications, and that is by affecting every aspect of drug metabolism or pharmacology in general, including absorption, metabolism, distribution, and excretion. So why am I interested in this somewhat esoteric topic? It's because drug efficacy and toxicity are regulated by non-modifiable factors such as age, sex, environment, genetics, ethnicity, physiology. These are things that you can't really change, although maybe you could change your physiology a little bit. And also your environment by moving to a different place. But in general, like for the most part, these factors that contribute to drug response are not modifiable. However, intestinal bacteria, which we know now can alter drug responses, are a modifiable factor. As we've heard about yesterday and through all the posters today, the intestinal microbiota are a modifiable aspect of drug response. So maybe we can understand this as a way to improve drug responses in people. So the systematic study of how intestinal bacteria, or just bacteria within our body, impact drug metabolism is a new and emerging field called pharmacomicrobiomics. It's a lot of syllables, but I think the name is very self-explanatory in which how microbes can impact the pharmacology of various medications. And this is, I'm going to just talk to you about a few key examples, which I think are very important and clinically relevant. So in the past several years, there's been a lot of emerging evidence that has systematically implicated specific bacterial enzyme functions in changing the efficacy of various medications. And one of the key examples of that is digoxin, which is a cardiac glycoside. A bacterium called Egertella lenta can reduce a single bond in the digoxin molecule, which I've highlighted with a star, to inactivate this molecule and make it and render it useless. Similarly, there's a class of enzymes called azoreductases that are really important for activating a molecule called sulfasalazine, which is critical for treating ulcerative colitis. These bacterial azoreductases will activate this medication and make it efficacious for usage in people. We also know that enzymes encoded by Helicobacter pylori can contribute to decreased bioavailability of levodopa, which is really critical for reducing the symptoms of Parkinson's disease. There's numerous other examples, such as increasing the GI toxicity or the nephrotoxicity of compounds like 5-fluorouracil, which is an anti-cancer compound, as well as acetaminophen or paracetamol, I think is what it's called in India. So gut bacterial enzymes can also compete for the same host enzymes that are critical for detoxifying some of these drugs. And by this competition, they render some of these medications more toxic to our body. So that's another mechanism by which gut bacteria can contribute to variable drug response. They can change the expression levels of things like the constitutive androstenedione receptor, which also is important for modulating susceptibility to anesthesia molecules, for instance. And a key example for folks who are interested in nutrition in the audience is the metabolism of choline found in things like eggs and meat into this molecule called TMA, which is then subsequently oxidized by host enzymes to create a molecule called TMAO, which is implicated in atherosclerosis. There's one more example of a mechanism in which gut bacterial enzymes can alter drug efficacy, and that's by actually changing metabolites that our body produces as a normal process of drug metabolism. So a number of hydrophobic molecules, not just medications, these can also be environmental toxicants or healthcare products like triclosan. These molecules are hydrophobic, and they are detoxified in our body in the liver by conjugation with a sugar molecule called glucuronic acid. And this is a small carbon sugar that, upon conjugation, renders this hydrophobic molecule to be hydrophilic, and so that it can be easily eliminated through urine or the fecal route. When these glucuronic conjugated molecules are eliminated through the gut, they encounter a class of bacterial enzymes called beta-glucuronidase, which have the opposite chemistry, in which they actually will hydrolyze glucuronic conjugates and reactivate them. So for the length of this talk, please remember that glucuronic conjugates in general are thought to be safe and non-toxic, whereas their reactivated counterparts basically are the parent drug, and these are often toxic in certain contexts, which I'll talk to you more about. So the reason I'm really interested in glucuronidation and the opposite process, deconjugation, is because a number of cancer drugs are detoxified by glucuronidation during phase 2 metabolism of these drugs. So this is a list of FDA-approved anti-cancer medications, and they comprise a number of different classes of drugs. There's tyrosine kinase inhibitors, HDAC inhibitors, some traditional cytotoxic molecules, and these are all basically conjugated with glucuronic acid in the liver and then excreted through bile into the GI tract. So what is beta-glucuronidase? This is a bacterial enzyme that is a non-essential enzyme in bacteria that's responsible for scavenging carbon. Remember, there's always a competition for nutrients in the gut. So this is a crystal structure of the E. coli beta-glucuronidase tetramer. Something to keep in mind is that we also have a mammalian homologue of this enzyme. And the lack of beta-glucuronidase in people leads to a lysosomal storage disease called SLY syndrome. So this is an essential enzyme in us. And whereas in bacteria, this is a non-essential enzyme that's responsible for carbon scavenging. And the reason we know it's non-essential is when you knock it out using CRISPR and create an isogenic mutant strain, the mutant in the blue line can grow at the same level of fitness as the wild type strain. Bacteria scavenge glucuronic acid, and through two biochemical transformations, shunt it into Krebs cycle to generate ATP. So this is a way in which bacteria that express beta-glucuronidase can derive an additional source of energy generation in the gut, where again, there's a competition for nutrients. Relevant for this talk, we can measure the rate of the deconjugation of a glucuronide molecule in vitro using a variety of bioanalytical tools, which I'll talk to you throughout the course of this talk. So here is a movie of the monomer of the E. coli beta-glucuronidase. So what you'll see deep within the enzyme, there's the catalytic site where the glucuronide moiety of the drug conjugate usually resides. And it's flanked by this red floppy loop that I think you guys can all see, also over Zoom. But this red feature is a catalytic site adjacent loop that almost acts like a clamp to tightly hold substrates close in the active site. The catalytic site of beta-glucuronidases are highly conserved and critical for their activity. To use an analogy of fishing, I used the catalytic site of beta-glucuronidase as a molecular bait to mine the fecal database of the Human Microbiome Project, which is a repository of 139 individuals and a collection of their microbiota from different body sites. So using this molecular fishing expedition, we found a variety of different structural features that are shared and also distinct between bacterial beta-glucuronidases. Through a lot of structural work that I don't have time to go into right now, we were able to find that there's overall 6 important key structural features that are shared by bacterial beta-glucuronidases in the gut. And we've given them extremely uncreative names. There's loop 1 gusses, which are in work that we've done in the last few years, indicates that they're the most important for reactivating small molecule drug glucuronides. There's also other types of gusses, uncreatively named loop 2, no loop, mini loop 1. Mini loop 1 and 2. Last year, we also discovered that there's 2 additional types of beta-glucuronidases, but for time's sake, I'm not going to go into them here. But we do know that they require flamin mononucleotide for their as an essential cofactor. So, what do we learn from doing all this molecular fishing? Well, we find that nearly every taxon that's represented in the gut has one of these 6 different flavors of beta-glucuronidases. So again, this is an enzyme that is found within most members of our microbiota. And because in biology, form fits function, these unique structural features tell us something about the underlying biology, and they indicate that each of these enzymes has a very distinct substrate. And the fact that these structural features, so these loop regions, are diverse, allows us to selectively target these loop regions, which again are actually absent in mammalian orthologs. So bacteria have loops next to their catalytic sites. Those loops allow us to selectively target bacterial beta-glucuronidases while leaving the mammalian orthologs untouched, which again, this is really important because their deficiency is can lead to severe congenital defects. So this is just a bar graph in which every one of these bars is one unique individual within the Human Microbiome Project's fecal database. And I've color-coded this figure for you, in which every one of these figures corresponds to a particular type of loop structural feature. And I think at a glance, we can see that different people have different distribution of beta-glucuronidases within their guts. And again, this probably accounts for some of the inter-individual variability that we're seeing in response to many medications. So again, to remind you, this is the list of anti-cancer medications that are detoxified by glucuronidation. And each of these medications, people respond differently to them. So, am I doing on time? 15 minutes, okay. So like I said, there's a great deal of inter-individual variability in response to cancer medications. And so we think that a reason for that is actually this variability in beta-glucuronidase enzymes. I told you earlier, although I speak very fast, you might not remember, I told you earlier that this red group, does this actually work? Is it working? Okay, yeah. So this red group of loop 1 gusses, that I think everybody can see very clearly, this is what kind of is the most different between each of these individuals, right? About 2/3 of people have loop 1 gusses, whereas 1/3 of people don't. And our molecular work in the last 5 years has uncovered that loop 1 gusses are what are important for reactivating drug glucuronide conjugates. And this includes a number of drugs that are on this list. So because we've hypothesized that inter-individual variability in drug response can be attributed to this differential proportion of beta-glucuronidases within people, we first wanted to find out what is the scope of the problem? Like how much of GI toxicity is actually attributable to variable deconjugation of drug glucuronide conjugates within people's guts. So Obviously, this kind of work is very hard to do in vivo. It's expensive and it's time consuming. And so we sought to develop a platform technology that could actually help us identify quickly and in a reproducible fashion what is the scope of epithelial damage that results from drug reactivation. So for this, I'm very grateful to the organ donors and recently deceased individuals from whom we've isolated different regions of the GI tract. So we can dissect portions of the GI tract from these donors, delaminate the entire tissue, isolate epithelial cells or any other cell type that we might be interested in, and establish them as organoids, which are 3D structures of these gut cells that we can grow in culture, or using a very specific stem cell factors, as well as other growth factors, we can convert these 3D structures into single cell monolayers that we can culture in the lab. These monolayers have an accessible apical as well as basal compartment, which makes them a very attractive platform to study drug metabolism and pharmacology in general. These cell platforms are also amenable to high throughput analysis, so we can very quickly measure things like viability, calcium signaling, redox stress, ER stress. We can also do more medium throughput assays, such as studying the integrity of the intestinal barrier, and also do more low throughput studies, such as quantifying cell swelling or measuring ER stress. So this is an image. Okay, so when we, sorry. So this is a very important platform technology because the way these cells grow in cell culture recapitulates how they actually grow within our gut. So within our gut, a single layer of cells separates us from the environment. And this layer of cells is a layer of polarized epithelial cells, which when these cells are grown in very particular growth conditions, these cells will polarize in the same way as they are in the gut. They are composed of highly proliferative cells, so we can maintain the stem cell aspect of the cells, as well as promote them to differentiate when we want to. These cells form very nice tight junctions. They allow us a way to measure barrier defects. And again, I want to just remind you, the reason why we're doing this is because GI toxicity is a really big burden when it comes to cancer treatments. So I'm going to tell you about an example of a medication called irinotecan, which is used for colorectal cancer as well as pancreatic cancer. It's also, its usage is also extending to things like triple negative breast cancer, for which there are very limited treatments available right now. So irinotecan is administered intravenously, where plasma carboxylesterase is converted into a molecule called SN38, which has a 10,000-fold higher potency of binding its cellular target, which happens to be topoisomerase. So topoisomerase is a molecule that's very important for unwinding DNA during DNA synthesis. And remember, the cell type in which there's tons of DNA synthesis happening is a cancer cell. So irinotecan is a really potent inhibitor of cancer cell proliferation. Another cell type that's highly proliferative is actually the gut cells. So for those of you who didn't know, our entire gut can renew every 5 days. So for that renewal to occur, we rely on a very robust pool of stem cells within the gut, and which also have a very high level of expression of irinotecan. Topoisomerase. So these stem cells we hypothesize are very susceptible to things like irinotecan and other cytotoxic agents. So SN38 is detoxified in the liver by liver-resident enzymes called UGTs to form this inactive SN38 glucuronide, or SN38G. When SN38G is eliminated via the bile into the gut, it's again encountered by bacterial beta-glucuronidases, which will deconjugate this molecule into the active SN38, which can exert cytotoxic effects upon the intestinal epithelial stem cells. In people, the side effects resulting from irinotecan manifest as bleeding as well as diarrhea. And often there's no way to really diminish this diarrhea because symptomatic treatment with things like loperamide has very limited effects. And the only way to actually relieve chemotherapy-induced diarrhea is to cease treatment. And this can actually lead to treatment, the progression of the cancer itself. So finding a way to treat chemotherapy-induced diarrhea is actually one of the main goals of my lab. And the way we're doing that is by targeting beta-glucuronidase. So by collaborating with a group of really bright mathematicians and pharmacologists at UNC in the Eshmelman School of Pharmacy, we've developed what we called a primordial physiology-based pharmacokinetic model in which we can actually estimate the amount of reactivated SN38 that we think is found in the gut following a single treatment with SN38. And we're using that concentration of SN38 as an example to expose this to monolayers of intestinal epithelial cells and examining what are the cellular sequelae of administration with this drug. And so just as an example, the concentrations that we find within the gut after SN38 treatment are between 10 and 100 micromolar. So when we expose intestinal epithelial monolayers to these two concentrations of drugs, we can measure correlates of cellular viability as well as epithelial integrity in vitro. And this is just a single snapshot of SN38, which again is the metabolite of irinotecan. Thanks. So I think anecdotally, we probably all know that, someone's family member had a particular cancer treatment and they did really well, whereas someone else's family member had a particular cancer treatment, the same cancer treatment, and they maybe were the same age, but those people fared poorer, like they maybe had, chemotherapy-induced diarrhea or mucositis, things like that. So what we've done is systematically, and we're in the process of doing, is systematically examining intestinal epithelial tissue isolated from people of various racial and ethnic backgrounds to examine how the same concentration of an individual chemotherapy can affect various correlates of cellular fitness. So stay tuned. It's not advancing. So I only have a very few minutes, but I just want to wind things up really quickly. In our culture, as well as many other cultures, talking about poop is a taboo topic. And so in a way to try to address this taboo and make it a little bit more palatable for us to talk about excrement in scientific terms, I've coined a term for studying excrement or fecal samples in a systematic and scientific manner, and it's called infemo. And the reason I did this is because when I wanted to figure out a simple biomarker for predicting who would respond poorly to chemotherapy-induced diarrhea and who wouldn't. And the basis of this is when animals are administered a single dose of irinotecan, we actually see an increase in the function of the bacterial enzyme beta-glucuronidase, which again is responsible for deconjugating those irinotecan metabolites. So we call this an in vivo assay. And the reason we're doing all of this is actually to figure out a way to inhibit chemotherapy-induced diarrhea. And we've done that by designing inhibitors that selectively target the bacterial beta-glucuronidase enzyme using that structural loop that I told you about earlier. In designing these inhibitors, we wanted to make sure that they had no impact on the efficacy of irinotecan. And we've done that using mouse models of triple-negative breast cancer, both immunodeficient and immunocompetent. This work is published if you would like more information. But ultimately, what we find is when we use these inhibitors of bacterial beta-glucuronidase, i.e. when we drug the bug, we are able to reduce chemotherapy-induced diarrhea in mice whilst ensuring that their tumor volumes regress to levels of irinotecan treatment alone. So this is a proof of principle that you can target selective bacterial enzyme functions to reduce drug-induced toxicities that are occurring, for example, with cancer treatments. So since then, we've moved beyond irinotecan and moved into mycophenolate and regorafenib, which are both other compounds used for stem cell transplant and liver cancer, respectively. We know that other drugs beyond just cancer drugs are also glucuronidated, and these are non-steroidal anti-inflammatory drugs. We have a paper coming out very soon, so stay tuned for that, in which we've shown that inhibiting bacterial beta-glucuronidase can also reduce the NSAID-induced enteropathy. So where do we want to go from here? Ultimately, we want to develop a clinical tool that will augment clinical decision-making. What we envision is collecting stool samples from people and incubating them with either authentic glucuronide conjugates or small reporter fluorogenic reporter molecules, quantifying the rate of beta-glucuronidase activity in an individual's stool sample and predicting that that person would fare either well or worse with a particular treatment. So right now we are collecting stool samples from individuals prior to and during treatment with irinotecan, during FOLFIRINOX treatment. And we're going to test the hypothesis that a higher rate of in vitro beta-glucuronidase activity correlates with higher amount of diarrhea, chemotherapy-induced diarrhea in these individuals. So ultimately, we want to parameterize the luminal reactivation of glucuronidated compounds and validate them in vitro and in vivo models, estimate the authentic luminal concentrations, and then use this as a way to guide ways to mitigate chemotherapy-induced diarrhea, either by use of things like, live biotherapeutics or medchem approaches to redesign the drugs while keeping the pharmacophore intact, or indeed by using inhibitors of the bacterial enzymes that impart this toxic activity. So the key takeaways of this, ultimately, I'll just skip to the key one, which is drug the bug, at least in theory, is an effective strategy to reduce chemotherapy-induced side effects in preclinical models and promises improvement of tolerability and efficacy of existing anti-cancer drugs. So my goal is to use microbiome targeting to advance precision medicine for all. And I would be remiss if I didn't thank, and most importantly, again, the organizers for this invitation and the Global Probiotics Council for arranging for me to travel here so I could tell you about my work in person. Thank you. I speak very fast, but here's my email address. Please feel free to get in touch anytime. So thank you.
Thank you very much. Thank you, Dr. Adria Bhatt, for giving us a detailed investigational data that you have developed in the laboratory. on an emerging topic, the role of the gut microbial enzymes on the some specific drugs. Thank you very much. So she will answer the questions at the end of the session. We will move on to the next online speaker. Yeah, he's connected. He's connected. Dr. Sanjay Kumar Banerjee is an associate professor in the biotechnology division of the National Institute of the Pharmaceutical Education and Research, Guwahati, India. His current research, Dr. Banerjee's research focuses to identify the novel targets and understand the molecular mechanism of the chronic and complex diseases like cardiac hypertrophy, heart failure, diabetes, and non-alcoholic fatty liver disease. His work revealed several novel cellular signaling pathways that perturbated during the disease progression. Over to Dr. Sanjay, please.
Okay, thank you very much for the introduction. Can you hear me?
Yes, sir, please.
Hello?
Yes, sir, please
Okay.
continue.
You're audible, but can you make it full screen?
Yeah, I'll go into full screen. Thank you. Okay. So first, I'd like to thanks to the organizer, especially Professor Ganguly and Dr. Nirja for inviting me and giving the talk. I want to be physically to be there, and due to my one small fracture in my leg, I'm not able to make it. But I hope I can like present online whatever I want to present physically there. So the title of my talk is Probiotics from Traditional Fermented Food from Northeast India, Impact on Cardiometabolic Disorder. So my laboratory last 15 years working on cardiovascular diseases along with diabetes and NAFLD. So whenever we look the diseases in human as well as making animal model, we realize that this not come as a single entity. Whenever try to make diabetes, the same the patient or the animals we got, they have also having a cardiovascular complication as well as they got fatty liver. So we realized that disease is come together most of the times, and it is a basically syndrome. So that's why my work basically now shifted to mostly on cardiometabolic disorder or cardiometabolic syndrome. And I'll talk a little bit about the probiotic work what I'm doing actually last I can say only 8 months. I am not in very new in this particular field. So as I talk about cardiometabolic syndrome, basically includes a cluster of conditions which include abdominal obesity, insulin resistance, and dyslipidemia and blood pressure. So when these diseases come, cardiometabolic diseases, it is not a single organ disease. It affects the heart definitely, as well as pancreas, liver, kidney. So those are together. Sometimes it comes with two disease. Sometimes come with three organ defects together. So very difficult to control or treat these kind of patients. So if you look actually the the statistics of cardiovascular disease in the world. So they are the #1 killer in the world. But India also not exception in it. In US, there every 34 seconds, 1% die in cardiovascular diseases. In India, burden also very much. If you look actually, the estimated annual death in India among all non-communicable disease, cardiovascular disease is the #1. 26% people die in cardiovascular disease. 1/3 of the senior citizens in India who are diagnosed with hypertension, 5.2% with a chronic heart disease, 2.7% with stroke. According to MCCD report 2008, they showed that cardiovascular accounted for more than 57%, actually more than 50% of the total death in the age group of 25 to 69 years. And India, the mean age of having the first myocardial infarction is 53, which is almost 10 years earlier than the happen in developed countries. Similarly, if you look at state-wise, Kerala is the ahead. They having a more number of cardiovascular patients and the death, and then followed by Goa and Andaman Nicobar. But if you look actually which state in India is the lowest events like Chhattisgarh, the Meghalaya and Nagaland. So this is very important to to work on it and to find how to prevent cardiovascular disease or cardiometabolic diseases. So if you look actually the therapeutic option, generally if the disease is very early stage, we generally having a preventive strategy. And if you come to the stage where there is preventive strategy along with the drug intervention, they work together. So when you look actually preventive strategy, small weight loss, moderate physical activity, dietary restriction in calorie intake, change in dietary composition, and also recently last 10 years, we talk a lot about if you improve your gut dysbiosis. So your gut health is very important for any diseases, not only like communicable diseases, but non-communicable diseases also play important role if your gut is good. And regarding the drug intervention, most of the drugs used for atherogenic dyslipidemia, elevated blood pressure, insulin resistance, those are the drugs always used when you go to the clinic. And my interest is that how to improve the cardiovascular health by giving a preventive measure. If you think about preventive measure, nowadays people talk about that probiotics. So what kind of bacteria present in your gut, they may improve your overall health. So, but how to improve it? So if you think about what kind of probiotics are important, a lot of research is going on. I'll talk today something which basically, in the culture of Northeast India, there are 8 states is there, and they use a huge amount of fermented foods. More than 300 fermented foods are used in different tribes and this area, and it is regularly they use. It's not like they use 1 and 2 days in a week. They do 2 to 3 times and regularly. So why gut and heart axis exist? Because we have seen a lot of literature that when you're having a healthy heart, you generally take a healthy diet. Your active lifestyle is very important, and you having a bacteria like Roseburia, Faecalibacterium, as well as your quantity of certain metabolites like TAMO, TMAs will be less, and also low LPS. But similarly, when you're having a poor diet or sedentary lifestyle, we have seen kind of presence of pathogenic bacteria like Klebsiella and Prevotella like this, and also having a high blood level of TAMO and TMA. LPS level also very high. So all this affect to the to the overall cardiovascular health, like you can get hypertension, endothelial dysfunction, developed of fibrosis, mostly in cardiac tissue. When they get fibrosis, they tend to go to the heart failure stage, and also you get a myocardial infarction or heart failure. So we know that there is a directly link to the gut dysbiosis as well as the cardiac health. So as I talk about Northeast India, as we having a very rich traditional culture, and they are still maintaining. If you look actually what are the most common food, fermented food use in Northeast, all 8 states are started from soybean, which is used a different part of the state with a different name. Then bamboo shoots, very important because this is very special part, and this Northeast they use the bamboo shoot. Then milk, fermented milk they use. Fermented fish, they use their fermented rice beer, legumes and cereals, and different vegetables also they do fermented and they use it. So this is a culture, and whoever comes here, they can see every local store, they sell this kind of fermented food. So my interest was that to explore one of the fermented food which already using here, and they having traditional knowledge, they having good health, especially cardiometabolic health. So the aim of a study to identify a probiotic strain from Northeast fermented bamboo shoots and validate the strain in animal model for efficacy in cardiometabolic diseases. Why I choose bamboo shoot? Because as I said, this is most of the states in the Northeast they use bamboo shoot. They having a different name, but the preparation also a little bit different. But good thing is that these bamboo shoots, they know that they provide a very high micronutrient. They boost immunity. And these bamboo shoots have the property to reduce body weight, reduce cholesterol, especially LDL cholesterol, and reduce blood glucose level. So, but there's not a lot of work has been done. There are some work is available, but people are not done in validation point of view, like any animal model as well as human studies. So if you look actually the, here in the map of Northeast, and if you see that these bamboo shoots actually used in different states like Nagaland, Manipur, Mizoram, Tripura, Meghalaya, Sikkim, Arunachal Pradesh with a different name. So what we basically did beginning that we looked at which part of the the states they hugely use this bamboo shoot fermented food, and they also having a good literature. So we searched that mesu is one of the preparation of bamboo shoot that obtained from Sikkim, and there are different kind of tribes like Lepcha tribes, Gorkha tribes, Limbo tribes. They use these bamboo shoots for their good health. So we thought we should work on the mesu, and we then started our work in the laboratory. So if you look the how they prepare the mesu in general, so there are three different kind of bamboo species available in Northeast. One is Bambusa tulla, Dendrocalamus hamiltoni, and Dendrocalamus sikkimensis. So there are three different species. So the preparation is very easy. Like they go for make the the bamboo shoots, and then defoliation of the bamboo shoot is the first step, and then you have to do cleaning, and then you have to cut a small pieces. Then you have to go for packing tightly into a glass jar, or mostly in a tribal people they put into the bamboo gum, and they keep for generally 7 to 15 days. But some places I have seen they keep for 30 days. So in that fermented bamboo shoots they use for different way in the food preparation. So, so my job was to how to make the same fermented food in laboratory condition, and to isolate different kind of lactic acid bacteria from this fermented food. So here actually I will give a small video and see that how we prepare in this bamboo shoots in our laboratory. So we collected from the local market these bamboo shoots. We cut into a defoliated the bamboo shoots, and then we cut a small round pieces, then remove the core part, as you can see. Then we wash in tap waters, and we having this bamboo gum. So and also one glass jar. So we cut a small pieces all these bamboo shoots and put into there in the glass jar first, and then put into this bamboo gum also. So we make a two different preparation in the laboratory. One is, and then put in the top of it the banana leaves. We tight it. This is the same way as they prepare in the tribal people. And now we put into this 30 degrees centigrade for 15 days for the fermentation part. So now I like to show the first two we prepare in the laboratory. This bamboo shoot, one from glass jar, another one from bamboo gum, and the other three we got it from Gangtok, like Sikkim. So the three the preparation we got from like commercially available bamboos bamboo shoot fermented food, and three are made by three different bamboo species. Like the third one, BT, they prepare from Bambusa tulla. DH prepare from Dendrocalamus hamiltoni, and the DS prepare from Dendrocalamus sikkimensis. So now I took two from the lab preparation and three from the commercial, and then we want to see what kind of lactobacillus is present in this 5 preparation. So that we have started the the preparation of the this bacterial isolation. So we make the small mesu homogenate into one small tube, then make a different dilution 10 to the minus 1 to 10 to the 9, and we put into the MRS agar plate. And after we getting these colonies, we collected them and stored into the glycerol stock. So this is one of the example, one of the preparation. We put different dilution. You can see that different colonies come. So one of the best dilution we choose so that we can get the very pure and isolated colonies. So now, as I said, that we having a 5 different preparation. So we try to collect as much as possible. Like we collected, 20 colonies from this, from this preparation. But last one BT, we didn't get a lot of colonies. One of the reason we realized that sometimes, in the bamboo shoot, when they ferment it at the end, they put some preservative that kills the bacteria. The last one, which also commercial stores, we not able to isolate a lot of colonies. Very less was that, but we collected few from it. So total 66, colonies were isolated. And then we characterize them for their in vitro property, whether they're having any probiotic properties. Like we started with morphological and also tolerance tests like shape, gram staining, acid tolerance, bile tolerance. Then we looked the cell surface property, hydrophobicity, autoaggregation, safety analysis, hemolytic activity, antibiotic susceptibility, physiological and biochemicals like motility, catalase, and adhesion property. And we also try to look the genotyping by doing 16S ribosomal sequencing. So, we want to identify all 66 isolates that we, collected from that, from 5 different preparation of bamboo, fermented food. So we isolate the, DNA, and then we send to at THST, Dr. Bhavatosda's lab for their, look that their sequencing by doing 60S ribosomal sequencing. And after completion of the sequencing, we come up with each and every isolates come up with their bacteria. Most of the cases they are 98% similarity, and then some of the color we are showing here, which looks different, blue color or kind of greenish color, they are kind of new strain which was not reported before. In bamboo shoots there are some work has been done, but there are new also species was found which is not found in the literature. So these are the all 11, like if you look the all 66, so they are some very common, except one of them we have not taken it, which looks like a pathogenic, but we took 11. Out of 11, 9 are lactobacillus strain, and 2 of them are Pediococcus and Wisela. So now we started to look all those bacterial species, whether it is reported previously with any kind of documentation with the cardiometabolic disease. So then we come up with one of the bacteria, Lactobacillus brevis, which is shown a very good literature search showing a very good data. So here basically we come up with one table, like looking only the few property like gram staining, morphology, catalase, motility test, and hemolytic assays. And when we put the table, and then from the table we come up with the 25 strain further from 66, which also have the all probiotic property, as well as they survive in acid tolerance test and bile tolerance test. So out of these, as I said, that NGBT36. So this is the strain, we take it forward for further validating in vivo model. And as I said, this strain basically have L. brevis is there. And we found that in literatures that previously there are some studies has been done that particular bacterial strain have adipogenic effect. They also have cholesterol lowering effect, also shown to suppress high-fat diet induce increase in body and epidermal fat weight gains. They attenuated several abnormal associated with metabolic dysfunction, inhibit intestinal absorption of cholesterol. So overall, this particular species looks very promising to go to testing in the next in vivo level. So this is the summary of their particular strain. They are catalase negative, they are gram positive, non-motile, and autoaggregation is 17.44%, hydrophobicity 14.81. Also, they have antimicrobial activity that done with the 3 bacteria, E. coli, Salmonella typhi, S. aureus. Antibiotic sensitivity, they are sensitive most of the cases except streptomycin, which is shows resistance. We look the literature also previously shown Lactobacillus brevis is resistance to streptomyces, and that hemolytic activity shows that this bacteria does not.
You have 3 minutes.
Okay, sure. And also look the gram negative, gram positive bacteria. This is a study to show that they are adhesion with the CACO2 cells. Now coming to the in vivo data, where we have just initiated the study. We are middle of it, but some of the interesting data I thought I can going to show you that the animal we use the C57BL6. Like total study was 14 weeks. The first 2 weeks we just keep them, you know, with a PBS and which is a control group. Then disease model also, you know, we keep the last 12 weeks choline deficient hyper diet, which causing basically fatty liver, insulin resistance, and cardiovascular complication. And the third group, which is a therapeutic model, we have given, you know, first 2 weeks every day L brevis, which is a 1 × 10⁸ CFU for 2 weeks, and then we continue, you know, with every third day lactobacillus brevis. And after, you know, 14 weeks, we're supposed to certify. We have completed till now 4 weeks, but we want to show you some of the data which may show some interest. And so in general, after 4 weeks, it's very difficult to look some data, but we did for the presentation that body weight gain was started from the CDHF group, which is a, you know, the cause maximum cardiometabolic disease, and there's a trend to decrease body weight after L brevis. Similarly, IPGT test was done 120 minutes, which supposed to be, you know, high value of the glucose, which was again, it's a very early to say insulin resistance, but there are tendencies that L brevis group having a lower glucose level. Similarly, we did some echocardiography study. Our institute has one image system, Vevo 3100, which works basically through ultrasound as well as photoacoustic system, which is a laser-based image system. And we did liver, fatty liver, you know, images, as well as we did some echocardiography study. I like to show the first in the fatty liver. We measured the fatty liver in by image system. We looked at the CDHF diet. There's an increase of fatty, you know, fat accumulation in the liver by doing liver echogenetic testing. As you know, the L brevis, there's a, you know, showing there's a decrease of the fatty liver. Similarly, we did echocardiography, and there are 3 different groups are there, and we did 2D echo to see that their contraction relaxation. And this is the summary of the, you know, study, that none of the you know, parameters are showing any significant change, but like to tell you that, you know, systolic volume was decreased in L brevis group, ejection fraction was slightly increased, fractional shortening was slightly increased. That's showing there's a tendency to increase, you know, that cardiac contraction. And I think with time we can know, generally CDHA model after 12 weeks, there's a, you know, there's a lot of decrease of the ejection fraction, fractional shortening. If this particular, you know, bacterial strain can increase the cardiac function, that will be benefit at the end stage. So this is a summary of a study that MESU is a fermented bamboo shoot. We prepare in the laboratory. 66 bacterial colonies were isolated and characterized by different in vitro methods. 11 bacterial species were identified by 16S ribosomal sequencing. Lactobacillus brevis was chosen for a further in vivo study in a mice model of cardiometabolic syndrome. Oral administration of Lactobacillus brevis showed tendency to reduce body weight, blood glucose level. Similarly, the same bacterial species showed tendency to reduce fat accumulation in liver and increase cardiac function. Till now, the data obtained very early stage shows very promising, but we have to wait for another, you know, few more weeks to get the final data, and a lot of histopathology, cytokines, all the parameters will tell me the, you know, the final outcome of this work. So what we wanted to say that we have done the, you know, we have,
Please
yes,
come to the conclusion.
this is the last slide. This is the last slide, sir. So finally, we want to see that this probiotic, whatever isolated, make a formulation, and if we can try in human. So, you know, I am looking some, you know, partners who are doing human studies and clinical studies. They look my data and how to take it forward to the human studies. So this is the acknowledgement. I am thankful to Dr. Bhavadas Das from THSTI who have done the sequencing study, and also different time point whenever I get any kind of, you know, suggestions or discussion, he give a lot of good input into the project. And 3 of the students who are involved into this project, Rasmita Datta, who is a second year postgraduate student who has done most of the study as I've shown the data. Siram Mahajan also part of the study. He has started the in vivo animal model. And Nabia she is very good on image systems. Most of the image data was developed by Nabia. So thank you very much, you know, for that, you know, the opportunity. Now I'll be happy if you have any questions.
Thank you, Dr. Sanjay, for sharing the interesting information from the Northeast region. Thank you very much. We will come back to you with a limited set of questions very shortly after the end of this session. We will move on to we'll move on to the next speaker. Okay. Yeah. The next speaker, Dr. Chittaranjan Yajanik. He's the professor and the current director, the diabetes unit of the KEM Hospital, Pune, India. He was the Helmut Moharat Award of the International Diabetes Toronto in 2009 and as Outstanding Investigator Award for the World India Diabetes Foundation in 2016 and the 41st Gopalan Oration Award Nutrition Society of the India in 2017 and 18 and has got many awards to his credit. Over to Dr. Chittaranjan Yajanik, please.
Thank you, chairpersons, and thank you, Nirja and Dr. Ganguly for inviting me. I'm going to speak about gut microbiota in diabetes mellitus and also add a little bit of sort of information on microbiome of B12 deficiency. I come from KEM Hospital, Pune, and this is the only diabetes department in the world where the logo is mother and baby because of our intergenerational and now transgenerational work on transmission of diabetes. We couple this work with modern day rapid lifestyle change. You can see here the modern Ganesh, whose lifestyle is very different than what we knew about in the past. I also represent two societies, the SNEHA India, which is Society for Natal Effects on Health of Adults, and International DOHAD Society, Developmental Origins of Health and Disease. With this, we have our efforts at primordial prevention, and the last talk was obviously very important and relevant to these ideas. I have to say I have been helped by Dr. Shrikant Bhutte, my PhD student who did work on diabetes in this presentation. In addition, the slide is not moving. Yeah. And I have to acknowledge help from many colleagues, starting with Yogesh, Saroj, and a whole lot of people with whom I have worked. Our funding has come from Indian agencies as well as agencies from abroad. And we are going to cover today broadly the diabetes microbiome concept, studies which led to it, Indian studies, especially in Pune and Chennai, some trials on probiotics for diabetes from the international literature, and the challenges and opportunities. This, of course, has been said again and again. There are lots of bacteria within and outside us. And there are lots of associations with a lot of conditions. And I think we really need to now sit, take a look, and decide where we need to make a further progress to make it etiological and related to the sort of causality. So the way I see is that in last 10 to 12 years, for a diabetologist like me, who knew about type 1 and type 2 diabetes, both characterized by high glucose, lipid abnormalities, autoimmunity in type 1 diabetes, insulin secretion defects and insulin resistance, and the driver for type 2 diabetes, the adiposity, we have a now new player, that's microbiome dysbiosis. This has been described in various studies, but we are struggling to understand whether it's a cause, consequence, or very likely both. The studies on diabetes and microbiome, there have been human as well as animal studies. And in last 15 years, if you just try to Google on the PubMed, there are more than 4,000 publications related to these. Most of the studies are cross-sectional and retrospective, which makes it difficult to understand causality. There are a number of other comorbidities which might add a confounding factor. There is also an effect of treatment, various supplements, and the pre and postbiotics, either taken as supplement or coming from diet, like the previous speaker suggested. So incisive mechanistic causality studies are still lacking. I'll just show one slide about type 1 diabetes. There is a big international effort. This is really new and we are learning, so it's very preliminary. The study is called TEDDY. It's driven from Sweden. And that has shown actually in type 1 diabetes, lot of population level heterogeneity of microbiome. It's coming mostly from white and non-Hispanic people. But overall impression there is that there is excess of streptococcus and lactococcus in the stool of patients with type 1 diabetes. And it's been linked with reduced butyrate production, reduced bile acid metabolism, and increased LPS. And we heard a brilliant talk by Dr. Bhatt, how these might be related to various suspected and unsuspected metabolic processes. The real hope for a clinician is that if we find a probiotic which might actually change the conversion from the preclinical to clinical type 1 diabetes, that would be a fantastic thing. But it's a dream and it's a long-term dream. There is a review on studies in type 2 diabetes, which summarize 42 studies of association of type 2 diabetes and microbiome. And the resounding message is that several diversity indices and the famous bacteroides to firmicutes ratio has very inconsistent associations with T2D. Let's take one like say alpha diversity. There are 13 reports of like say Shannon type of analysis, no association in 9, positive association in 2, and negative association in 2. If you go one by one, it's the same story for different microbiome sort of components. Though broadly, we can say Bifidobacterium, Bacteroides, Faecalibacterium, Akkermansia, and Roseburia are negatively associated, while these Ruminococcus, Fusobacterium, and Glaucia are positively related. Lactobacillus seems to be of great interest, especially in India, but the associations are inconsistent across the sites. And that is shown pictorially here for the different, like say, phyla or whatever you call it. Here they have a, code that negative studies are shown in blue, positive in red, and you can see for the same bacteria, there are sometimes both types of associations. In other studies and other population, it may be only one-sided, and in animals, it might be very different. Overall, to say that this phylogenetic tree and its components have a very different association in different studies, and therefore it's not possible to make an overwhelming positive or negative statement. What I really feel the message there is that we need population-specific markers if we are to actually make a progress in this. And this, of course, takes us in the realm of what is a buzzword today, either precision medicine or personalized medicine. And towards the end of the talk, we might see what might be the future designs of the study. But this should be a central issue that the patients and the controls should come from the same population. They should be matched in other confounding factors. Then only you are going to be able to make some sense of these associations. If we now look at Indian diabetic patients, like the previous speaker said, I mean, Indians have a heightened genetic predisposition But more important than that, and especially for research which has happened in my department over last 40 years, we have shown a resounding role for intrauterine undernutrition and subsequent relative overnutrition, so-called mismatch hypothesis, which is a driver of epigenetic programming of diabetes in Indians. This leads to an earlier age at diagnosis at a much lower BMI. The environmental factors which precipitate it are rapid economic transition, rural to urban migration, and now we have the very dubious distinction of being the world's capital of diabetes with estimated more than 100 million patients in India. Indians are different for microbiome, that is these biases, as well as the phenotype. And my department has contributed quite a lot to definition of phenotype of Indian diabetic patients. As I said, there is genetic diversity, there is endogamy, there are diverse dietary habits and geographic locations, even for differences within India. And I actually described the Indian phenotype, which we called thin fat Indian phenotype or the thrifty phenotype, where we showed that for a low BMI, we have higher body fat percent, we are more centrally obese and more dysmetabolic compared to the Europeans. And 40 years ago, this was a one-man, one-control study, myself and my registrar in Oxford. And in one study, we could find major differences in all these factors. For the microbiome, the group I work with, Yogesh Shauche's group, written by Shrikant Bhute, there are clear differences at the family level distribution of the bacterial taxa if we compare, like, say, Indian, Bangladeshi, American, Spanish, and Korean, with a suggestion that within the Indian subcontinent, there are more similarities compared to the other parts of the world. If we were to look at the overlap of OTUs at 97% similarity, only 3 OTUs are common to all these populations in the studies of type 2 diabetes. And if we look at distinct OTUs for other populations, they are to the tune of 100 to 200. For Indians, they are more than 450. So we have a much more diverse microbiome than the other populations. If you do the principal component, then Indians, of course, stand out very different than the Americans. And if you look at patients and controls, what this slide shows is that Indians have even more diversity within our groups also. So that adds to the complexity of associating the microbiome with the phenotype. And we need a consortium and very specialized studies with a lot of thinking to make sense out of this. What are the studies from India on microbiome? 2 studies from Pune. The first one from my department, Shrikant Bhute. Then again, his sort of department with Gaike as the first author. And 2 studies in an Indo-Swedish collaboration between Dr. Mohan's unit in Chennai and Copenhagen. Their numbers are bigger because we had very small grants at that time. And the specialty of our study was that the samples came from one of the 3 prospective birth cohort biobank that I have. So we had ready-made samples in biobank, which were serially collected over last 30 years. And in the Pune children's study, in the parents of the children, we were able to find 19 healthy subjects, 14 newly diagnosed diabetes subjects, and remaining known diabetic patients. This allowed us to have actually their extensive phenotype over years and a sample of stool whereby we could do the microbiome. And Shrikant's special approach to this under Yogesh guidance was that we looked not only at bacterial diversity, but we also had special primers for archaea, fungal, and eukaryotic microbiome. And that actually makes it very distinct from other studies because we were able to find differences across the board. And what were the findings? We had a reduction in alpha diversity in patients with high glucose, whether newly diagnosed or established, compared to the normal glucose tolerant people. If we look at microbial dysbiosis, there were a number of phyla like Bacteroides, Firmicutes, Proteobacteria, Actino, etc., where there were differences between the populations. If we look at mean abundance, 20 discriminative OTUs showed a characteristic transition from the normal glucose tolerant to early or newly diagnosed patients to the late or already diagnosed patients, suggesting that as the metabolic abnormality increases, the differences become more pronounced. And then we actually look at differences of, like, say, total bacteria. They were much less in the diabetic patients compared to the NGTs. Similarly for Bacteroides, but for Lactobacillus, it was exactly opposite, that the diversity, or sorry, the absolute count was much more for Lactobacillus in the diabetic patients compared to the other two groups. Again, showing how complex it is. The principal components again showed the three groups stood out different, but on further analysis, we find that there is actually diversity across the group, which is not restored after treatment of diabetes. In other words, it continues to remain with the patient. And this is a cartoon which Dr. Bhutte drew, which summarizes all these findings. And for me, I have to just summarize that actually our impression was that the microbiome dysbiosis which we were observing could be a consequence rather than cause of the high glucose. And this was because, like, say, with higher glucose, there were more disturbances. But equally, we found some evidence that as a dysbiotic state is reached, it itself affects the gut wall integrity, causes endotoxemia and chronic inflammation, and exaggerates the two basic deficiencies or basic defects in diabetes, namely insulin resistance and deficiency. So then it becomes a self-propagating sort of phenomenon. If we look at other study from Pune, where they added one more group of pre-diabetes to the normal and the two groups of diabetes which we described, the findings are broadly similar. And here, the pre-diabetes and NGT are closer to each other than they are with the higher glucose, again suggesting a dose response type of phenomenon. This study additionally measured circulating metabolome and showed very interesting findings in relation to different metabolic sort of domains like oxidant stress, inflammation, amino acid metabolism. But interesting to show that the different components of microbiome have very different associations with the same molecule in the blood. And therefore, I think this provides a rich database for future investigation to see which of these components can be picked out.
In 2 minutes?
Maybe a little bit more. I had negotiated with Dr. Nirja because she asked me to give B12 also.
Oh, okay. Sure.
Sorry. So in other words, we found a lot of similarity between two Pune studies, and that actually could be a geographical thing. Then comes the studies from Chennai, which are in collaboration with the Danish group. Here we have two groups from India and two groups from Danish, normal glucose tolerant and pre-diabetes. And very interesting impression you get by looking at this heat map is that there are more differences between the countries than between the two metabolic groups. And this is again one more indication that there is great diversity across geographical areas, and therefore we need strong local controls to make sense out of it. And same thing at taxonomy level and at function level. Therefore, what they said was they found 16 OTUs which were enriched in normoglycemic and 144 in prediabetes. They were actually separately additional gut microbiome signatures when prediabetes was discovered. Several Prevotella OTUs were enriched in prediabetic while Megaspera in IGT. But interestingly, these are very much enriched even in healthy Indian individuals. The pro-diabetic patients had higher pro-inflammatory state, and these were different than the Pune studies, again highlighting the differences. Then second paper from that group reports in type 2 diabetes. And what interested me here was that they extend the findings, but they say that T2D related microbial changes at taxonomic level were seen only in the Danish population, not in India. So again, more food for thought. Things are getting quite complicated. One of the reasons which I have actually for studying serial birth cohorts is that we have demonstrated that metabolic abnormalities in adult age can be tracked back to very early age, like 6 years of age. Here, those who are pre-diabetic at 18 years were already pre-diabetic at 12 and 6 years. And they already had metabolic abnormalities which lead to it. In other words, if you study only adults, you get only an end-stage cross-sectional picture. We really need to go into early life. Now, what about interventions? This is a meta-analysis reported again by Indian workers, Dr. Paul and his colleagues, where they had 58 studies with more than 3,500 people which took part. The studies were extremely heterogeneous, but there was a common finding in the meta-regression that with with the probiotic treatment, there was a fall in fasting plasma glucose, HbA1c, fasting insulin, and HOMA-insulin resistance. This tells us that it might be very useful, of course, to pursue this, but we have to be very aware of the confounding factors like age, BMI, baseline markers, dosage, intervention duration, and the study region. But the hope is that there is hope. So I now think that we are moving around in a vicious circle. We don't know which comes first, chicken or eggs. So there is dysmetabolism, dysbiosis, which drives the whole cycle of exaggerated inflammation, dysmetabolism, complication, and adverse outcomes. And where to enter this cycle would be interesting investigation. We have published genetics of subgroups of diabetes in India. And what I would like to point your attention with type 2 diabetes and its subtype, the moderately obese subtype, in Indians, but not in the Swedish, giving us some indication that B12 might be involved. You know, 30 years I have worked on B12 and folate and found a lot of influence on fetal programming and programming of diabetes. So this was a very rewarding research. And then we did B12 deficiency microbiome studies. Lots of collaborators and students, but especially colleagues from UK, from University of Kent and Quadram Institute. And what we find is that there is a variance of microbiome in the B12 deficient and B12 sufficient people, but it's also substantially influenced by the age of the person. so that in children, that's boys and girls, and fathers and mothers, the differences are more than within the B12 deficient and sufficient groups. Then we looked at the phyla differences and found that there are differences in firmicutes, actinobacteria, proteobacteria, and Bacteroides and cyanobacteria. And if we go to the next level, then we can see quite distinct differences in relative abundance of a number of genuses. And to me, this is the first demonstration from within India that the genera of the microbiome associated with B12 deficiency are different. They are influenced by the age of the patient, sex of the patient, and also the vitamin B12 status. Some of these different bacteria are actually producers of B12, while others are consumers of B12. And the hope of this analysis, which we are of course continuing to make it more refined, is that if we are going to be able to improve the population's B12 status, we will have to create probiotics with more of B12 producers and less of B12 consumers. Is there any other evidence for this? And I'm thankful to my colleagues from Tata Consultancy Services, Krishna, and I think Pina, who is attending this meeting. He has shown this slide, I think, in his presentation, that they did a sort of a meta-analysis of microbiome in different populations, related it with the average BS in these relations as available from the literature. And what they find is mean abundance of vitamin B12 producing bacteria decreases as the B12 deficiency increases, while the B12 consumers increase as the deficiency increases. So it must be a dysbiosis between producers and consumers that might reflect at least part of the B12 status of the host. So for future, I think we need a much larger number of participants in our study. We need and locals. We need prospective rather than cross-sectional studies. We need functional microbiome analysis, including ultra-deep metagenomic and preferably metatranscriptomic sequencing, combined with metabolomic assessments, not only of the bacteria, but also of the host. And for host, we need genetics, lifestyle, and multiomics. I am actually trained in epidemiology, where Mendelian randomization is now assuming great significance in trying to prove causality in different studies, in addition to human intervention, which has its own limitations because of the cost involved, the designs involved, and a whole lot of things. So I think we should think about Mendelian randomization. And ultimate hope will be to have clinically translatable products. These ideas were told to us by an economist. Many of you perhaps know. This is my last slide.
Professor Young.
Yeah, this is the last slide, that we need to keep Bradford Hill's octet in mind for future studies in the microbiome. Thank you very much.
Thank you, Professor Yagnik. Sorry to interrupt you in a couple of times because the program had only 20 minutes for you.
Thank you.
Okay. And now we will have the question answer session. So I request Adra to come on stage. Sabu and I will coordinate this. So the questions are for all the three speakers of this session. And I would ask the audience offline first to ask the questions.
I have a question for Dr. Ardra, so it's an interesting model that you talked about with the notobiotic mice showing changes with the glucuronidase. Now, the thing with the cancer drugs is that some of them affect the small bowel and some of them affect the large bowel. Checkpoint inhibitors, for instance, would cause colitis. The irinotecan that you talked about and maybe mycophenolate would cause small bowel diarrhea. The question really is it bacteria in the duodenum which produce beta-glucuronidase which are important, or are they colonic beta-glucuronidase producers which are important?
Thank you for your question. That's very astute of you. We have yet to undertake a specific and systematic biogeographical examination of beta-glucuronidases in the gut. I do think that their overall intrahepatic circulation, recirculation probably contributes to some of duodenal, small intestinal damage as well as colonic damage. So yeah, I don't really have a clear answer for you.
Actually,
Yes.
Adra, we need to have this answer because as Dr. Yagnik was saying, that we need to intervene when they're 5 to 6 year old children. And TAMIS and KEM is doing a study where they are moving with intervention at this stage. So a lot of misery could be. So emanative, sudanitab, dasanetab, when they are given for the chronic myeloid leukemia, they move as a whenever there is a blast formation, you move the new drug and then you move the new drug. So if we could intervene in the gas sector and with ICANN or whatever you have and identify them early while they are being treated, we can perhaps avoid use of the consequent drugs.
Absolutely.
So this for both Dr. Yagnik and you, that the best method will be to intervene them at a period when you could do something, because if you look at the Yagnik study, they, even when they have been treated for diabetes and controlled it, the microbiome is remaining same. It is not changing, and the metabolomes are also same. So if you intervene, you have to continuously intervene to them, which is very, very difficult. So why not intervene very early into this? And to two very small questions. One to Dr. Yagnik, that you have followed these girls for nearly 40 years. And if you have the data, have they changed the data? Because your data showed at one time that if the epigenetic signature didn't change, so they went on producing the same challenged babies. So does the microbiome changes in these years in them, or it remains same? And Dr. Sanjay Banerjee, I have a question for you, that there's very interesting, but if you are looking at one of the species of the lactobacillus, Baustosh has got a huge number of signatures of the same species. So you need to really find out more about these signatures, these sequences which you have, and you have chosen NGT6 or 26. I've forgotten the number at the moment. But if you choose these signatures, which are very important signatures to look at the metabolome and other changes which are happening there, the study will become richer. Thank you.
So we'll start. You want to? No. Okay. Dr. Yagnik?
Okay, thank you, Professor Ganguly. As you know, we conducted world's first primordial trial in adolescents in our cohort to improve the health of the next generation. So that's even before the childhood. And that is an intergenerational influence. And we already have results of the neurocognitive improvement in the children by providing only 2 micrograms of B12 to the adolescent mothers from the time of adolescence till the day of the delivery. We are now following up these children between 6 to 10 years of age to see if we have changed the cardiometabolic risk. And that study should be over probably next year. So I'll be able to tell you more. What we have observed is that the children born in the trial have about 250 grams higher birth weight compared to their mothers. They have larger head circumference, they have larger abdominal circumference, but very interestingly, they do not have excess adiposity. So I think whatever we have done over years by way of general improvement in the background due to advance or development in the nation, plus our intervention seems to help us to come out of the thin fat phenotype. The definite answer will come a little later. My microbial biobank started only about 10 years ago, because before that we had no idea about this. So we have blood samples. So if we can find microbial signatures in the plasma and serum, and I know there are, then we can go back 2 generations and find whether it has changed. And I seek your support in obtaining funding for this. Okay.
Banerjee.
Thank you, sir.
Banerjee, Dr. Banerjee.
Yes, thanks, Dr. Ganguly, regarding your interest. See, I'm also very new in these topics, and after coming to Assam, I realized that in Northeast there is a huge amount of fermented food, and my research started, exploring them. So yes, one of the species we have started working on it, and we don't know that actually whether this is different than other similar species from other strain. So I sent the bacteria to Dr. Bhavata's lab to go to the whole genome study to look at how they are different from others. That is important. And what I said, we are going to do this. Also, we send the fermented food to Bhavata's lab to do the metagenomic study. Whatever we have isolated, it is still present into the bamboo shoots or not. So he'll isolate the bacteria, whatever present in the bamboo shoots, he can look the presence. So which will give me confirmation whether whatever we isolated, that is also present. And whatever you said, very interesting that we should look the little bit on metabolomics point of view. If after 14 weeks, if we really see there's a reduction of insulin resistance, and also I can see there's a tendency to reduce body weight, which was previously reported that adiposity, anti-adiposity effect is there with the lactobacillus brevis. So if it's there, I think it's interesting that see that how we can, take it, more forward, go for more, study. Like what is the probiotic property? There are some ICMR guidelines, but that guideline is, very, it is not that they done everything. They're having a 5 and 6 some property they want to do in vitro. But if you really think that is a very good effect preclinical, I'll come to you, sir, and I need a discussion what to do, what to take it forward. So, and I'm really excited in the field, and a lot of hope is there. So thank you very much for giving me the opportunity.
Dr. Sanjay, can I suggest one more thing, measurement in your samples?
Yes, sir.
Because vitamin B12 is made only by microbes in nature.
Yes, yes.
And if you also measure B12 and 1-carbon metabolites in your brew, I think
Yes,
that might give entirely additional viewpoint on what might be actually useful in another way. Because B12 deficiency is quite rare in the Northeast.
Yes.
Right.
So, sir, I think after your lecture, I realized actually, that vitamin, B12's, source basically, a lot of different bacteria which consume, and some of them are producing. So those are very important things to look into it. And I think you might know that in Northeast, I know how much microbiome work is done, and our Northeast signature will be completely different than other part of India. And here cardiovascular disease is not that big, but overall, cardiometabolic disease are there. but they are somewhat protective from, as I said, Nagaland, Meghalaya, they having a less cardiovascular disease compared to other states. So we really need to explore actually this area, how they are different.
I have set up a birth cohort and an adult cohort in the Dibrugarh Assam Medical College. It is
Sure, sir.
biobank.
Okay, sir, someday I have
And
to
you
discuss
can
more.
actually benefit.
Dr. Bhatt, this is based on your presentation, I have a concern.
Yes.
And it is when we do phase 3 drug clinical trials in different ethnic groups and different populations, we are factoring in the variation that exists in the drug metabolizing cytochrome P450 kind of enzymes. But we never ever thought about the microbiome altering the safety or even the toxicity of the drugs across population. Now, this is a big issue, especially when you're raised on the anti-cancer drugs. And we never studied this at all.
Thank you so much for preaching to the choir. Thank you for bringing that up. That's definitely the reason why we're doing this work, because the importance of bacterial drug metabolism is largely understudied. This is a very new and developing field. And like you're saying, you don't see what this does until you actually do the work in people. And at that point, we've spent $1 billion on developing a compound that ultimately ends up in the trash, right? Because of too many side effects. So this is why I think this work is incredibly important, because we've developed, mathematical models to estimate how much drug is actually reactivated in the gut by bacteria. For just one enzyme, beta-glucuronidase, there's millions of other enzymes that we don't even know about. Well, maybe not millions, but thousands at least, that might be involved in metabolizing drug intermediates. For example, glucuronidation is not the only method of detoxification, right? There's also sulfation. So sulfatases are, bacterial sulfatases are a largely understudied group of enzymes. So yeah, you're absolutely right. This is a critically, this has been an overlooked aspect of drug development and drug metabolism. And thank you so much for highlighting the importance of what we're doing. I appreciate it.
Yeah, Dr. Adra,
Can I say one thing can I say one thing in diabetes? Metformin is known to affect microbiome, and microbiome is known to affect metformin metabolism and efficacy. So that has been known for some time.
Dr. Adra, nice talk. Just curious, is there any evidence to know how quickly after birth are these bacterial glucuronidase expressed?
Wow. I've actually never thought about that.
Yeah.
But we do know that a number of lactobacilli express beta-glucuronidases. I mean, the availability of so many birth cohorts would be a great way to actually examine the development of what we call the GUSome, or the beta-glucuronidase -ome. We call it the GUSome. I think those will be really great. We have fantastic bioinformatic pipelines to actually quickly determine GUSes based on actual authentic structural data that we have. So that would be a great thing to do.
Yeah, the only reason I ask is because in conditions like kernicterus, where it's supposed to be a problem with the beta-glucuronidase not being active at birth, and having to treat it so that it is activated and the bile is conjugated and excreted out. So is the problem that beta-glucuronidase is not active, or the bacterial glucuronidase is overactive?
What was the disorder you spoke of?
It's called kernicterus. It is
right. So I moved rather fast, but so there's very specific structural features that we've found that contribute to substrate specificity amongst bacterial GUSes. That particular structural feature is absent in the mammalian homologue of GUSes. So because in biochemistry and in biology, form fits function, there's a very distinct substrate preference for bacterial GUSes versus mammalian GUSes. So from what I know about the story, it seems like the mammalian gusses are more important for the bilirubin recycling and not bacterial gusses. Bacterial gusses, like I've showed you, they either work on small molecule glucuronides or endobiotic glucuronides, such as certain hormones and neurotransmitters, or on really large molecules like heparins and certain large glycosaminoglycans. So,
Dr. Bhatt, can I offer you a collaboration? We
Oh.
have serial samples
yeah.
on the stool samples from the children born in our third generation, starting with meconium and every 3 months for first 2 years along with mother sample.
I look forward to chatting with you offline.
Okay,
That sounds
thanks.
amazing.
Yeah.
Thank you.
Adira, that was a lovely talk and
Thank you.
it's a lot of good research. I was only thinking about the idea that whether the point would drive the point home that usage of probiotics would be undermined. It would be less in use, where we are trying very hard to push probiotics in the larger interest, global interest, for betterment. So it was just a thought because we have not researched on that topic
Sure.
at all.
I propose my work as a complementary approach to using probiotics or live biotherapeutics or symbiotics or things like that. This is a complementary approach to that, which I believe, which I agree is invaluable for improving health.
Yeah, I think there is no more questions from the audience, but we have a few questions from the chat box also. So we are running out of the time. I think we'll go ahead with the chat box questions very fast. The question is from Professor Prakash Halami, and the first question is to Dr. Sanjay Banerjee. Does the consumption of the fermented food, such as the bamboo shoot, is credited with a lower risk of cardiometabolic disease? Is there any specific evidence available for this? Dr. Sanjay Banerjee.
Yes, yes. So there is no specific evidence till now. These are all belief on through traditional knowledge that whoever consume traditional food like this kind of bamboo shoot every day, so they having a better health. But there are actually some identification of bacteria from this bamboo shoot are done, and they suggest that because they have a good lactobacillus bacteria and that might be benefit, but this is the indirect kind of evidence, not a direct evidence. So we are trying to do that. We want to do the preclinical study where we're giving the bamboo shoot itself to the animal, as well as that probiotic, all those bacteria we have isolated, we can give, and parallel see that possible is that bamboo shoot itself has a lot of good metabolites or flavone or all those present fiber. Those also may be beneficial effect. So you have to do some study, then only I can answer these questions.
Okay. Then we will move on to the next question from Dr. Vimala. The question is to Professor Chittaranjan. The question, what is the impact of the diabetes medication on the microbial dysbiosis?
Okay. I mean, metformin was the first drug where it was shown to actually affect the gut microbiome of the patient. And there is a lot of literature on this. Other drugs which might be acting, I don't think they have been studied systematically. But for metformin, there is a big story.
Yeah, we will have one more question. That is to the Dr. Meera, to Dr. Adira. But my question, the question is do you believe there is uniformity in the bacterial population in the cancer patients you studied?
Wonderful question and very astute. No, I think people's microbiomes differ, and they particularly differ based on your diet, geography, as a lot of the work in the last 2 days has shown. However, what's really interesting about bacterial beta-glucuronidase that I don't have a lot of time to talk to you about in detail is that it's an enzyme whose expression is subject to catabolite repression, which means that This enzyme is expressed by bacteria, the Enterobacteriaceae in particular. It's upregulated up to 5,000-fold upon the presence of a glucuronide substrate. So in work that we've done so far, it's pretty evident that the presence of a medication that is glucuronidated within, a mouse model, for instance, does upregulate the expression of beta-glucuronidases. So again, this allows us to develop a targetable and a druggable enzyme and function.
Okay, thank you. As we are running out of the time, we have already answered so many questions from the chat box, but we don't have enough time to answer all this. So this will be answered through the online mode very shortly. So I think we will stop this session here. Thank you very much. Thank you all the participants participating here in the audience and as well as in the online mode. I also take this opportunity to thank the organizers for putting one such mega event in the southern part of the country, in the state of Kerala, in the extreme south of India. Thank you very much, especially by hosting the RGCB Director, Professor Chandrabas Narayana. This is very important event that is happening for the first time in the state, especially in the context of the initiation of a new dedicated center, the Center of Excellence in Microbiome in the state. That is what we are just mentioned this, in association with the Rajiv Gandhi Center for Biotechnology. So thank you very much. Thank you, organizers. And also for giving me an opportunity to co-chair this fantastic meeting. Thank you. Thank you all.
Thank you, Sabu. I think he's going to let us felicitate, right? No, your mic is on.
So thank you so much. So now it's time for the felicitation. So I request the chairpersons to felicitate Dr. Adra Bhatt. So thank you so much, Dr. Adra Bhatt. So I request to take a snap together. Yeah. So now it's time to felicitate our chairpersons. For that, I would like to welcome Dr. Siad and Dr. Rajesh onto the dais. You also will get one now. So thank you so much, Dr. Sabu Thomas. Thank you so much, Professor Chandrabas Narayana. Thank you all. Thank you, Dr. Siad and Dr. Rajesh. All right, so we are moving on to the next scientific session, that is scientific session number 4, role of gut microbiota beyond intestinal health. I would like to welcome the chairpersons onto the dais, Professor Ajay Bhala and Professor Jagmeet Madan. And I request our chairpersons to introduce the keynote speakers.
Hello. So very good morning to one and all. At the outset, let me thank Professor Ganguly, Nirja, and the entire organizing team to put up such a brilliant scientific program. I think very, very impressive, very engaging, I think, and we all have had an academic feast from yesterday. till today. I also want to highlight the triple connection, which has brought nutrition on the forefront, along with gut microbiome and probiotics. And I think this symposium has given a huge opportunity for different stakeholders to come together. It's the nutritionists, food microbiologists, food technologists, medical practitioners to have a crosstalk. And it's time that, this kind of interdisciplinary engagement should happen for better and applied work to come in future. Just one statement as a nutritionist, I wish to place on record, and whatever I've heard from yesterday, that in a country like India, being the developing country and now likely to be a developed nation, I think we have to vouch for preventive nutrition than curative nutrition. And when we talk of preventive nutrition, somewhere there is a commitment towards the quality of diet in everyday food. And with today's nutrition science emerging, precision nutrition can come in everyday foods. And we can connect that with the outcome in terms of the microbial diversity. And like it is very clearly said that, if we are healthy, we definitely have more of microbial diversity than when we get diseased. So it's better that we prevent ourselves from becoming diseased, and that's the role a nutritionist of this country has to play. So with these words, I think I'm going to invite our first keynote speaker, Professor George Chandy. Yes, sir. Professor is a director, BCMCH, Tiruvalla, Kerala, former director, CMC Vellore, Tamil Nadu, India. He's an internationally renowned medical researcher with special interest in gut microbiome, hepatitis B infection, preventive strategies in acute fatty liver of pregnancy. He's a fellow of Royal College of Physicians, Edinburgh, and many international hospital society. And I think his innovative model is replicated in smaller hospitals, extending its impact on underserved communities. We look forward to your presentation, sir. He's going to be talking on I think, it is. Talking on gut microbiome and the clinical revolution, the widening horizon. Thank you, sir.
I want to especially thank Professor Ganguly for the amazing leadership that he shows in the area of science, and particularly from CMC over 20 years ago, the way he helped us when we took baby steps in the area of antimicrobial stewardship, as well as the appropriate use of antibiotics. Today, I want to thank Professor Ganguly and Dr. Nirja for this very special opportunity to be at this fantastic symposium. To Professor Chandra Bas and Dr. Srikumar and Dr. Sabu for the excellent arrangements made. And as we said a little earlier, the fact that we can be here together at such an amazing scientific symposium is such a privilege. I've been given the task of speaking about the gut microbiome and the clinical revolution. Dr. Nirja emphasized that I must bring in, as a practicing clinician, all the aspects that we need to look at. On the 20th of July, 1969, Neil Armstrong, when he landed on the moon, said this: "One small step for man, one giant leap for mankind." So, respected chairpersons and ladies and gentlemen, I believe that metagenomics and all the other things that we have talked about is perhaps for the scientist a small step, but for the world, it is going to be a huge, giant leap forward. Elie Meshnikov, the father of probiotics and the Nobel laureate, said this: "The majority of diseases begin in the digestive system when good bacteria are no more able to control the bad." This is the corollary in society as well. When the bad controls the good, society becomes sick. Professor Ramakrishna and myself worked at the Christian Medical College, Vellore, where our mentor, Dr. Martin, used to say that we own the rotten gut. At that time, I didn't quite understand it till the Australian team led by Shane Warne landed up in Bombay, and he came down with a Bombay belly. And I thank God that we have all these bacteria within. As a practicing gastroenterologist and hepatologist, I've always known that it is the high bacterial load in the gut that leads my patients to trouble, especially when they have decompensated cirrhosis. And I knew that the ammonia bypasses the liver because of the portosystemic shunting and affects the brain. In my practice, 60% of patients have gas, and again, the small intestinal bacterial overgrowth has been one of the major problems, and I've been using VSL-3 quite a bit. And the wonder drug is rifaximin, which is a non-systemic antimicrobial agent, believed to be a gut cleanser. And unlike other antibiotics, rifaximin increases the alpha and beta diversity and is a eubiotic, as shown in this graph, which was exemplified in the RIFSIS trial. So coming to looking at microbiota in gastroenterology, what are the well-established systems that we have? We know about the enteric infections. We know that H. pylori causes ulcer. We know about Clostridium difficile and the antibiotic-associated diarrhea. We also know about the biliary and pancreatic sepsis. Roles postulated for microbiota in gastroenterology include diverticulitis, inflammatory bowel disease, both ulcerative colitis and Crohn's disease, as well as celiac disease and CA colon. What about other diseases and microbiota? A whole lot of diseases we've been talking about yesterday and today: Parkinson's, Alzheimer's, and neurology, depression and anxiety, as we heard from our Japanese colleagues, cancers, and autoimmune disorders. So the butterflies that we felt in the stomach that we always talk about, or the gut feeling, why is it that we talk about the gut feeling and not about the brain feeling or the heart feeling? And today we are proud to say as gastroenterologists that the gut is our second brain. So we knew all along about the gut-brain axis. So when a patient comes to us who are tense and had problems, we know that there is a connect. But today we know much more about the 3D axis. So I just want to, it's a busy slide, but I just want to highlight this: the microbiota gut-brain axis, the endocrine axis, microbiota gut-heart axis, with the kidney, the whole immune process, the liver, and the lung. Over the urbanization, civilization timelines, it is interesting to note that the scientific milestones followed this. So from our natural habitat, we built our environment, while our gut microbiome also evolved. So today, we talk about the built environment microbiome. And we know that the microbial communities, along with the metabolites, result in a whole lot of chronic diseases, inflammatory diseases, bronchial asthma, whole lot of psychiatric stress-related conditions. And we also know that over a period of time, as infections dropped, the chronic diseases, the immune disorders tended to increase. So I'm grateful to friends in AIIMS Bhubaneswar, in CMC Vellore, and my own team at the Central Research Laboratory in Tiruvalla for their increasing interest in this area. So how do we look at the role of microbiota in clinical disease dynamics? This schematic representation by Vijay et al. In the European Journal of Clinical Nutrition, where he spoke about the role of gut microbiome in chronic diseases. It's a busy slide, but it shows you how the gut microbiome, the gut microbial diversity, and the gut-derived metabolites with all the ones that we know leading to a host of diseases, or we think that there is a definite relationship with these diseases. My area of interest is the liver and changes in the gut microbiota. The dysbiosis with systemic inflammation is involved with a host of diseases, certainly the chronic hepatitis, alcoholic liver disease, and we had a beautiful presentation yesterday, liver cirrhosis and hepatocellular carcinoma. The prevalence of fatty liver, as was mentioned yesterday, is only increased 30% across the country, 50% in South India. And our aim, our hope, is there some way we can prevent progression? So when a patient comes to me with fatty liver, we grade them grade 2, grade 3 and all that. And then I try and explain to them what fat does to the liver. When there is an injury, the body tries to respond and there is cicatrization, there is fibrosis. How do we reverse this process? And we don't have any drugs for it. We now have called it metabolic dysfunction associated fatty liver disease, but we are looking for help in this area. In non-alcoholic steatohepatitis, we have a buildup of liver fat, and we know that the firmicutes bacteroides ratio, there is a positive correlation with steatosis and fibrosis. When bile acids are essential for fat solubilization, in the presence of dysbiosis, bile acids are preserved. But that leads to increased permeability of the gut microbiota and increased microbial growth. And in the presence of oxidative stress, the pathogens associated molecular patterns, the PAMs, there is further inflammation and there is liver disease progression. All this is proven data. So when I look at the liver and the microbiota, I find that when there is short-chain fatty acids, acetate, propionate, and butyrate, that can prevent liver progression. Can we go on to precision medicine, personalized medicine, make that happen? How can we prevent the gut microbiome-derived metabolites, mainly the TMAO, from worsening the situation with NASH, fibrosis, onto cirrhosis? So our friends in All India Institute of Medical Sciences, Bhubaneswar, did this study looking at patients with lean NAFLD and obese NAFLD, and they found that there was a relative abundance of certain organisms in each category as depicted in these plots. This is unpublished data, but they are working on this, looking at the microbiome diversity, clinical features in NAFLD to try and see whether we can tweak the gut microbiome to help in this difficult situation. There are a whole lot of other studies with probiotics in NAFLD, which we need to look at. I want to look at the heart with you. We had a little presentation earlier from Assam. The trimethylamine leads to trimethylamine N oxide, resulting in increased blood pressure, cytotoxic effects on the cardiomyocytes. And there is a suggestion that TMAO in blood can be used as a cardiovascular risk assessment. So there was a study done by Wang et al., published in Nature many years ago, 10 years ago, where they looked at 1,800 stable patients. All of them were patients for elective coronary angiogram. And the TMAO levels were measured along with cardiovascular disease, plaque size, and they found that the TMAO levels had a 2.5-fold increased risk of a major adverse cardiovascular event. This was independent of the traditional cardiac risk factors. So at a time today, when we hear about a lot of sudden deaths in young people, I wonder whether this study that was done can be replicated to try and see, because we don't have any markers. We have tropi when the patient has come with a heart attack. But when a patient comes to you, how do you assess the cardiovascular risk? COVID-19 taught us a lot. At that time, we didn't know very much, but we knew that there was a tremendous immune response. We also knew that the cytokine storm led to lung injury. A lot of them were admitted. So at that time, several studies came up, which are interesting data for us to follow up. Fecal microbiota transplant in rats resulted in a decreased expression of inflammatory factors, decreased acute pulmonary injury, and reduced risk in COVID-19 progression. So when you look at germ-free mice, you find very poor immune response. So also with malnutrition, with microbial dysbiosis, there is progression of age-related respiratory diseases as against eubiosis. You restore eubiosis of the intestinal microbial community. There's enough evidence to show that you age in a healthy way. Whole lot of other lung conditions we need to look at to prevent progression of asthma and lung inflammation in chronic obstructive pulmonary disease. Two very interesting factors for the practicing gynecologists. The gut microbiota, the diversity and function, plays a very important role in gestational balance, because with decreased bacterial diversity, there is a preponderance to developing gestational diabetes. And my wife is a gynecologist, is very excited about this. And the possibility of preeclampsia, again, happens when there is decreased by bacterial diversity. So could this be an early marker for gestational diabetes and preeclampsia? Endometriosis, the gynecologist struggles with chronic pain and infertility. Gut microbiota have favorable effects on clinical, immune, and physiological parameters, and patients benefit from treatment with probiotics containing Saccharomyces boulardii. Some of my nephrology colleagues, I've seen them using probiotics left, right, and center. So I was suggesting that this team, we need to discuss something and find answers for this. Are they doing the right thing? I do not know, but they know that in stage 5 chronic kidney disease, there's increased Neisseria, Lactinoclostridium, Bifidobacterium, and they know that that worsens the kidney symptoms. And so they are using probiotics quite a lot. Precision medicine and personalized medicine is certainly the answer. Looking at metabolic disorders, we know that dysbiosis leads to poor regulation of fat storage, obesity, metabolic disorders. So fat excess and high sugar intake leads to colonization of enterobacteria, production of genotoxin colibactin, and the pro-tumorigenic activity. And so we have this new metabolo-infectious triad of microbiota dysbiosis, colonization of enterobacteria, and metabolic diseases. We talked a little bit about cancer. So gut microbiota end products affect intestinal coverage, encourages carcinogenesis. And we know that Helicobacter pylori causes cancer of the stomach. We know that genotoxic colibactin producing E. coli, the COP-EC, is involved most probably with colorectal cancer. Increased bacteroides in carcinoma prostate, and in CA breast, in the breast tissue, we find increased bacillus and decreased lactobacillus. The gut microbiome helps definitely in reducing the risk through anti-carcinogenic metabolites and short-chain fatty acids, while there are a host of other metabolites that increases the risk of cancer disease. The gut-brain axis is an area of great study. There is the proteopathy. There is this term called mapranosis, microbiota-associated proteopathy and neuroinflammation. Inflammaging, which is the prion hypothesis of neurogenerative disorders. So my colleagues looked at patients with Parkinson's disease, and in their study, they found that there was a significant decrease in Lactobacillus ruminus. In the 15 patients they studied with Parkinson's disease, they had taken their spouses, 15 spouses as their controls and normal controls as well. Significant reduction in the presence of Lactobacillus ruminus in these patients. We need to study this much more. Immunological problems, rheumatoid arthritis. It is known that there's an increased abundance of Prevotella copri and a decreased abundance of Faecalibacterium. So we definitely know that this gut microbiota dysbiosis and chronic inflammation is bidirectional. Amit Datta et al. In a study in CMC did the characterization of Crohn's disease patients, as you can see in this taxonomy bar and heat tree. And they found that in active Crohn's, there was lowered diversity, while healthy patients as well as patients with Crohn's, with stable Crohn's, had much better diversity. As seen in this heat map, the abundance of patients with acute active CD as against compared with stable and healthy patients is very evident. So again, my gastroenterology colleagues are using Bifidobacterium probiotic a lot. Is that the right way or not? It is for the scientific community to tell us. The salivary microbiome it's been shown that the microbiota and circadian oscillations and the changes that occurs due to age, coffee, tea consumption, and the microorganisms could be associated with tumorigenesis. And there are lots of correlative studies with the 16S rRNA sequencing reiterating these findings. So the periodontal bacteria, a whole lot of them, can be associated with carcinogenesis. So there are three mechanisms that have been postulated for cancer by oral microbiota: bacterial stimulation leading to chronic inflammation, the influence of cell proliferation, and the production of carcinogens. The skin-gut axis, a whole lot of studies on this, leading dysbiosis associated with psoriasis, skin cancer, and the like. We had a beautiful talk by Dr. Adra Bhatt on pharmacomicrobiomics. So I wondered, when she made that presentation, why is it that we clinicians never asked ourselves, when we give the same drug to two patients, the response is very different. Very, very exciting area. Whole lot of things that we need to do. Is it the microbiome that is doing all this? Very exciting times ahead for us. So to summarize, the microbiome gut organ axis is a huge big area we need to try and connect this. And again, precision medicine will be the answer. So the way forward towards a clinical revolution would be to predict, to analyze, but at the same time, to intervene. How do you predict? There are 4 stages. The stage 1 is a compositional and functional characterization. Stage 2 is data-driven hypothesis generation. Stage 3 is experimental validation of the hypothesis. And stage 4 is the development, therapeutic development, and clinical research. This is what we've got to work towards. And as we predict, you have 2 approaches, the bottom-up approach or the top-down approach. And in doing that, you have the DNA-based approaches, the metagenomics. You have the RNA-based approaches, which are the metatranscriptomics, protein-based approaches, which is metaproteomics, and the metabolite-based approaches, the metabolomics. So you can choose to do the bottom-up approach or the top-down approach. Could start with the metagenomics or with, and I would prefer starting with the metabolomics to get us answers. And to intervene, we have the prebiotics, the probiotics, the symbiotics, the combination of synergistic prebiotics and probiotics, and the postbiotics, which are metabolized, produced by the beneficial bacteria to support health. In this study done by the AIMS Bhubaneswar group, Balamurugan et al. Had HIV patients on antiretroviral therapy, on appropriate therapy, and he fed them probiotic chapati. And he found that it decreased the viral load and improved the CD4 counts. This is unpublished data which he's working on. Several studies on probiotics in GI disorders, probiotics in cancer, and probiotics in metabolic disorders, which we need to look at in great detail. And we need to manipulate the gut microbiome for better health with probiotics, with engineered microbiome, with the fecal transplant, which we're going to hear about, and a whole lot of other strategies with therapeutic options. So to conclude, we have something very exciting. And I said, it's as good as landing on the moon, because we now have characterized quite a bit. We need to do much more. We know a lot about the composition. We need to learn a little more. We need to look at the functions clearly and the signatures, as Professor Ganguly said, and intervene with dietary modifications, prebiotics, probiotics, postbiotics, the transplant, and the bioengineering. So this clinical revolution, I think, is at a phase of takeoff. And we have huge widening horizons ahead of us. And that's why we are here. Thank you, Professor Ganguly, Nija. Thank you so much for this opportunity. Thank you for your patient listening.
Thank you, Professor Chandy. I really liked what you said. Gut is the second brain. And you talked about the gut-heart axis. You know, on the lighter side, we always knew they used to tell women that the way to a man's heart is through his stomach. But you found scientific reason now here. Thank you so much. We will move on to the second talk. We'll have the question and answer sessions in the end of the session. Inflammatory bowel disease is something which is very little understood. There's like confusion about the etiology. Management of inflammatory bowel disease is something which varies from country to country. Our country probably follows a different pattern. There are new drugs coming in. But the most exciting part is about fecal microbiota transplant. And we have with us Professor Vineet Ahuja, who's a professor of gastroenterology at All India Institute of Medical Sciences. He's also the Associate Dean of Clinical Research, and he's a very dear friend. So he will be highlighting the role of fecal microbiota transplant in inflammatory bowel disease. Professor Vineet Ahuja.
Thank you, Chairpersons. Good morning, everyone. First of all, I'm grateful to Professor Ganguly and Dr. Nija Ajela for giving me the opportunity to come out here. And I would also like to congratulate Professor Ganguly for what I would call like science stewardship or leadership in terms of getting all the young minds out there and continuing to enthuse them by getting them exposed to the best in science. Thank you so much, sir, for this great initiative, sir. I'll be talking about microbiome manipulation. Thank you. So I'll be talking about microbiome manipulation therapies in IBD, and we have had some very exciting talks since morning, and every talk has taught us so much. Now I'll start with an example out here. For example, just to tell you that how science uncovers certain things and how things which seem to be of common sense may not be true. Like what are these? These are bears. Like you had a great talk by Dr. Yajnik, then you had a wonderful talk by Professor Chandy. And so if you were to, as students of science, if you were to take bear as an animal model, what kind of disease would you take the bear as animal model for? Diabetes, fatty liver, something like that, metabolic syndrome. You would all agree All of you would agree for that? Yes or no? Yes. Well, bears don't get diabetes. In fact, recent research shows that hibernating bears could hold a clue to treating diabetes. They probably have 8 key proteins, which have been found that they hibernate. They have great BMI and etc., but they never develop metabolic syndrome and diabetes. So this is how baffling things can be. And inflammatory bowel disease is another example of a disease which can be very baffling. It is a disease of unknown etiology, chronic immunoinflammatory disease comprising of ulcerative colitis and Crohn's disease, where you get ulcers in the small intestine or the large intestine. It affects people in the prime of the youth and unfortunately stays with them all through their life. And as yet, we have not been able to find a cure for inflammatory bowel disease. A lot of research has gone into this field. And if you look back over the last 2 decades, you find a plethora of drugs which have come up. Monoclonal antibodies, oral small molecules, anti-TNF agents, lymphocyte trafficking inhibitors, IL-23 inhibitors, IL-12-23 inhibitors, JAK inhibitors. And yet at the same time, we find that all these drugs, they have what we call as a therapeutic ceiling. They act, but they lose their response soon after. They don't act in more than 30%, 50%, or 60% of cases. You don't get 100% of response, and at the same time, till the time patients are taking these drugs, the patients have a response. The moment the patients stop taking these drugs, majority of them lose their response. So obviously, there's a big issue out here. So why does it happen? Like in the sense that why? Because most of these drugs, they are acting on their immune modifying, and we knew that there is a lot of redundancy in immune circuits. So probably that if you're blocking one hub, the immune mechanisms are going through another hub and again exciting inflammation. So if you go back to the drawing board, what you would do, you would go back to the pathogenesis of disease and see whether you can act on it or in a different possible manner. If you look at possible culprits for inflammatory bowel disease, we find that there's a genetic susceptibility, there may be altered epithelial cell responses, aberrant immune responses, and again, another very important component, that is environmental factors, of which diet and gut microbiome could be a principal player. So do we have a gap in knowledge out here? Can we attack the microbiome out here? And can it change the course of the disease? That is the question. So we obviously have 4 generations of gut microbiome manipulation. We have the antibiotics, the diets, the prebiotics, the probiotics, fecal microbial transplantation, and also postbiotics, as Dr. Chandy showed you in a slide. Postbiotics are also coming up. So what is fecal microbiota transplantation? It is essentially a restorative approach. In probiotics, you're giving one strain, some billion spores and all. In FMT, you're throwing everything in, all the bacteria, all the viruses, all the fungi, all the archaea, and it is infusion of a healthy suspension from a healthy individual into the GI tract of an individual with the disease, with the hope that it would refurbish the microbiome out there. And if it refurbishes the microbiome, it resets the immune system and possibly changes the course of the disease. So I'm not going to show you any animal experiments, mice experiments. I'll just show you a simple real life animal experiment, in the sense that this is published in Gut, which is a top rated journal in gastroenterology circles. So this is kangaroos. Kangaroos, they have small babies, which are known as zoes. So zoes, when they get orphaned, because if the mother kangaroo is poached and all, these zoes are often exposed to conditions which predisposes them to diarrhea, etc. So Cynthia Ranch, who was a game rancher, what she did was that she took up the poop from healthy kangaroo babies, and subsequently she made what are known as the microbiota poo shades, and does the video run? Okay, if it doesn't run, I'll leave it. Yes, it runs fine. So what I did was that she took healthy zoes, took their poo, that is the fecal matter, mixed them with milk, lactose-free milk, and fed it to the young kangaroo babies who were very sick, who had a lot of diarrhea. And she had thousands such cases where all of them improved, clearly showing that there is some wisdom in this approach and this can work. So now let me come to the first question, that before you're going to change the gut microbiome, you have to first decide whether it is the cause or the effect. Previous speakers have talked about it, and I've told you that possibly we are not very clear whether it is a cause. Let's see in IBD. In inflammatory bowel disease, if you look at gut microbiome changes, you find that compared to other healthy adults or compared to diseases like amoebic colitis, there's a different kind of microbiome which is seen in inflammatory bowel disease. This pattern was seen in North India. This pattern is also seen in South India by CMC in CMC Vellore by Professor Ramakrishna, who have shown, yes, there is a distinct signature in patients with inflammatory bowel disease. But that does not answer the question whether it is a cause or effect. Similarly, in inflammatory bowel disease, you may have patients who are in remission, mild activity, patients who are very sick, and you look at the gut microbiome signatures, you find that there's a difference in gut microbiome signature in patients who are doing well compared to patients who are very sick or who have very severe disease. But again, this could just well be the effect. Then what we also picked up was we picked up healthy people. Let's say people who are living in rural areas, people who are living in urban areas, then people who are living in Ladakh, because that is a place where we have set up health camp every year, and we have never seen a case of inflammatory bowel disease out there from last 2 decades, just to look at the gut microbiome. And what was surprising was that urban people, they had a worse microbiome as compared to rural people or the lay people. So if you collect all this information together, you find there's a stairway. Acute severe ulcerative colitis, worst microbiome. Ulcerative colitis, slightly better than them. Urban healthy, normal, but inferior to rural healthy. Rural healthy, slightly inferior to high altitude healthy. Again, it does not show that there is a cause or an effect, but it tells you there's a lot of association out there. And if there is a lot of association out here, it seems reasonable to manipulate the microbiome. So comes up fecal microbial transplantation, where essentially you take donor feces, mix them, blend them, filter them, and with the help of endoscopy or oral capsules, you infuse them into the person with GI disease. We often use most of the times we use a colonoscopic approach, but sometimes in patients, particularly with liver disease, what has been seen that the upper GI approach or putting it into the duodenum, which is the first part of the small intestine also, you can do fecal microbiota transplantation. A component of this is a donor, and donor needs to be very, very healthy. It is not healthy in the regular sense that, okay, I'm a healthy person. No, the donor has to be extensively screened because you may end up transmitting some disease. So the donor has to go through a very rigorous questionnaire in terms of travel history, in terms of infectious history, in terms of drug history, in terms of various exposures, and also various kinds of testing, so as to be very sure that the donor is healthy. In fact, even if somebody is healthy but has a BMI more than 25, we do not take up that person as a donor for fecal microbiota transplantation for the theoretical risk that you may end up transmitting metabolic disease, etc. So these are, if the videos work, these are the ways we do fecal microbiota transplantation at our center. We have been doing it since 2014. And FMT is not restricted only to GI diseases. It has been seen in various non-GI diseases. Also, a lot of data is coming up where it has been shown that it is effective in autism, Parkinsonism, schizophrenia, Alzheimer's, even in augmenting immunotherapy for cancer responses. So it has a whole lot of audience. It is something like which is probably becoming a movement. So I come to my next part of the talk now, where I would tell you about our experiences in terms of changing the microbiome. And so, microbiome manipulation can be done with the help of fecal microbiota transplantation. It can also obviously be done with the help of diet, because diet would change the microbiome. So the research hypothesis was that in patients with ulcerative colitis, which I told you is a kind of inflammatory bowel disease, if you do FMT, along with that, you give the different kind of diet, an anti-inflammatory diet. Can you induce remission? And subsequently, for the next 1 year, can you maintain the remission? So we did a randomized controlled trial and published it, which was fecal microbiota transplantation with anti-inflammatory diet, followed by anti-inflammatory diet alone. And it was found to be effective in inducing and maintaining remission over 1 year in patients with mild to moderate ulcerative colitis. Essentially, the trial design was that patients of ulcerative colitis were randomized into 2 groups. One group continued the standard medical therapy. These were patients with mild to moderate disease activity. The other group, 7 sessions of fecal microbiota transplantation were given over an 8-week period. Along with that, the diet was also changed. They were given anti-inflammatory diet. After 8 weeks of FMT, anti-inflammatory diet was continued for a period of 1 year, and we looked at clinical responses, looked at endoscopic or mucosal healing, and also differences in fecal calprotectin, which is a marker of inflammation. The anti-inflammatory diet which we gave, the principles were to avoid lactose. Yes, you can give curd and buttermilk, to avoid gluten-based grains, to avoid processed food, canned food, red meat, and encourage plenty of fresh fruits and vegetables. The clinical outcomes showed that at 8 weeks, as well as at 48 weeks, there was a significantly better response in patients who were given FMT and anti-inflammatory diet. And even the endoscopic responses, that is mucosal healing, was superior in patients who were given this kind of therapy, suggesting that this is proof of concept. In a way, this therapy does work. Obviously, it needs to be validated in much larger trials also. And the interesting thing was there was no major adverse event. And secondly, obviously, if you're going to do a cost-effective analysis, it is going to be much cheaper than giving the monoclonal antibodies, where the cost can range from 2.5 lakhs to 6 lakh rupees an year. So the next part is that Professor Chandy showed you that, okay, you have a concept. Subsequently, you would like to characterize it, do a compositional characterization, functional characterization, then build up a hypothesis, then take it to another animal model, test it out, and then go to postbiotics. Let me see that if we can complete the story out here. So these are my two PhD students, Manaswani and Aditya, who have done this very nice work, where all these patients who received FMT, they took the samples, the fecal samples, as well as mucosal samples from them, looked at the gut microbiome and also the fungus, the gut microbiome, and also looked at metabolites, because metabolites are more important than microbiome. And also looked at the same patients when they had got a response to FMT, what happened to the microbiome, and metabolome. And as I told you, they looked at two sites, the mucosa-associated microbiota as well as the fecal microbiota. So at both the sites, I'm not going to go through the bacteria, but just to show you broadly, yellow is the UC circle, that is pre-FMT. Pink is the post-FMT. See the diversity, richness, composition, how it has changed. And it has become so much near the controls. And it has happened at both sites, both at the mucosa as well as the fecal. Similarly, we see similar kind of trend with metabolites. Metabolites seem to normalize, move towards a level of controls, the kind of composition of metabolites which control population had after patients had received FMT. Again, you see fecal metabolites as well as colonic tissue metabolites showing a very good response after FMT. So this is concluded that yes, there is a change, that is a possible mechanism. There's a change in bacteria. There's also a change in the metabolite that helps. So the next step was that you're showing it at fecal level as well as mucosal level. Is it possible that there's another niche which is more conserved, and possibly that is a master regulator? You know, like stem cells are the master regulators. It's like somebody who's close to the prime minister possibly has more chances to influence, something like that. So there is another population out there which is known as the crypt out here. Do crypt have bacteria? Do those bacteria possibly have a more influence? So the next part of the study was that fecal bacteria have been looked at, mucosal bacteria have been looked at. Let's look at crypt bacteria. And so it took a lot of effort to get those bacteria from there because they're very sparse in number. You had to do fish. Thereafter, you have to do laser capture microdissection, and thereafter do the 16S. And clearly, again, it showed that even crypt-associated microbacteria, there has a completely different population. They're more aerobes. They're not direct facultative anaerobes or obligate anaerobes, because obviously the oxygen concentration is higher out there. There are aerobes out there, and those aerobes also, there was a different population in patients with ulcerative colitis, which changed after fecal microbiota transplantation, showing that, yes, the benefit comes out there also. So the mechanisms were, there's a change in microbiota and microbiome at fecal level, mucosal level. There's a change in microbiota-derived metabolites following intervention at fecal level as well as mucosal level. And there's also a change in crypt-specific microbiome following intervention. We couldn't do the metabolites at crypt because of the very sparse material which comes out from there. So this part of the story is completed. You've done a clinical trial showing that in a disease, ulcerative colitis, randomized clinical trial showing the benefit of FMT and anti-inflammatory diet, establishing FMT as a therapy. Thereafter, you have looked at microbiome changes, metabolite changes at 3 sites, and of this, from the heat maps, we picked up a very promising signal for urolithin. Urolithin, which is found in pomegranates, etc., and it was seen that it was very much increased after FMT, particularly in patients who are responding. So obviously, the next part of the trial was that, okay, the hypothesis has been built up. And as Dr. Chandy showed you, now is the time to go back to animal studies. So what kind of model are we going to build up out here? We decided, let's look at the big organoid lab, and in organoids, test out whether these metabolites work or not. Organoids, as you all know, are 3D culture systems. So again, instead of mice model, where obviously the genes would be different, here these are patient-derived organoids, where you take biopsies from colonic biopsies, and subsequently you make the 3D spheres of them, spheroid cultures. And so organoids were set up from non-IBD controls, as well as ulcerative colitis patients. The organoids were treated with urolithin, which is a metabolite, for interest for us, so as to look for the changes. And for changes, we did single cell transcriptomics to find out whether there is any decrease in inflammation or not, what happens in various cell populations. So these are the various organoids which are growing, and they were all enriched in the intestinal stem cell population. Subsequently, urolithin was added to them, and it was seen that with the help of urolithin, when single cell transcriptomics was done, essentially what was seen was there was a decrease in the inflammatory population, which was very much present out there in patients who were treated with urolithin, patients of ulcerative colitis who were treated with urolithin. So all the three processes have been completed till now, and now we are looking at forming postbiotics in terms of looking at urolithin-based therapies and how we can go and in terms of making a capsule out of them, capsulized form of them, and whether that can be useful or not, or making a cocktail of metabolites and finding that it is useful or not. And finally, another interesting point out here, this is for all the people sitting out here who want to work in this field. There's one question which is absolutely unanswered out here, and whoever answers this question obviously is going to make a major contribution to this field. And that is the single most important factor in all of this, is that some donors somehow have a profound effect in terms of getting the success for the endpoints. In terms of some donors have a profound effect on the microbiome when for the disease population. So these are what we call as super donors. So how do you identify the super donors? What kind of microbiome population do they have? What kind of diversity do they have? And I think that's a key question which needs to be answered. And finally, just 2 slides I have, since this is in Kerala, and there's a recent study which came out from our group, and this is essentially a coconut water. This is a dietary change, and how simple things can influence the microbiome, can influence the disease activity. It is not always that you need to go in for most expensive things. So this trial showed that coconut water was given to patients with mild to moderate ulcerative colitis, and it induced clinical remission. It did not induce endoscopic remission. So in the sense that it gave a marginal benefit, not a very major benefit as you would expect with steroids, but at the same time, again, something which can be easily practiced. It was a well-conducted trial, a rigorous trial in terms of it was a randomized, blinded, placebo-controlled trial where patients of mild to moderate ulcerative colitis were given for 8 weeks. 400 ml of coconut water was given. Why? Because we thought it's a good source of potassium, and previous data from our group and THSA, Dr. Amit Awasthi's group had worked on it and shown in mice model that when you provide potassium, it decreases inflammation as compared to sodium, which increases inflammation. So when you provide a source of potassium, you give coconut water for 8 weeks. So it is like adding a good dietary addendum. It was seen the patients achieved clinical remission, clinical response, and fecal calprotectin also fell down. Endoscopic responses were not seen as were seen with fecal microbiota transplantation. Interestingly, there were a lot of changes in microbiome, which we also saw when patients were given coconut water. So this is what I would say a proof of concept study. Again, obviously requires validation in a larger trial, but provides first of its proof, which shows that 8 weeks clinical remission and response was significantly higher in patients who received coconut water compared to placebo. And coconut water increased the relative abundance of bacterial genera, which negatively correlated with clinical and endoscopic disease activity in patients with mild to moderate ulcerative colitis. With this, I end my talk, and thank you so much. This is our IBD group, and these are collaborators from THST, RCB, and ICG. Thank you so much.
Thank you. Thank you, Dr. Vineet. I think you have really stimulated the young minds here, and we are extremely thankful for you to have shared your elegant detailed research in such a meticulous manner. And I'm sure all of us, including me and many of us sitting in the audience, have a lot of questions which we will take after the next presentations. Thank you very much. I invite on stage the next speaker, Dr. Souvik Mukherjee. Dr. Souvik is the Assistant Professor, Biomedical Genomics Unit, National Institute of Biomedical Genomics, Kolkata, India. His interest has been in host metagenome interactions in human health and disease, population genetics and molecular evolution of host pathogen interacting genes, wastewater surveillance for detection of SARS-CoV-2 and antimicrobial resistance genes in sewage water. He has published his work in high-impact peer-reviewed journals, and he's today going to enlighten us on host microbiome interactions in the development of chronic inflammatory disease in humans. Welcome, Dr. Souvik. We look forward to your presentation. Okay. Okay, so I stand corrected. He is the Associate Professor in Biomedical Genomics Unit, National Institute of Biomedical Genomics, Kolkata.
Hi, thank you all. Thank you for this introduction. And I thank Professor Ganguly to inspire me all the time and to do my research, and that I'm standing here and presenting the work, the confidence instilled is by him. So I work in various types of diseases, and most of them are based on skin diseases. So this is a platform for the gut microbiome, but I will give you an overview why studying the skin microbiome is also important. So I come from NIBMG, National Institute of Biomedical Genomics. It's in West Bengal. And there we study not only the microbiome, but also the host factors. Host factor means host genomics and epigenomics, and how to stratify patients based on the risk alleles and then compare their microbiome. Well, so microbiome, we all know that. So since I come from NIBMG, I have to skip these slides because audience are mostly fond of the human genome. So I need to say them that you see, after the human genome, microbiome can be considered as our second genome. And when the human microbiome project started, it was considered that the 90% of the cells in our body is microbial, although now that stands corrected, and it's now that 50% of the cells in the human body are from the microbiome. And we are fond of doing GWASs, genome-wide association studies, but now is the time of MWASs, metagenome-wide association study. And this is that microbiome is now all pervading, present in different parts of the body. And host microbiome interactions, we mean that microbiome, individual genetic background, the food habits, they maintain a homeostasis of health, and any perturbations in this can lead to a disease. Well, the human microbiome has also been assessed for answering the missing heritability problem in the complex non-communicable diseases now. So during the time of GWASs, there was a GWAS gold rush. Everybody was doing GWAS with all types of diseases and finding many associations. Now this has shifted to microbiome, and microbiome has now been associated with a large number of diseases. You find Nature and Science cover stories, and full issues are now dedicated to it. However, healthy microbiome is not same for all. And when we say healthy microbiome is not same for all, it is true that healthy genome is also not same for all. But the extent of the changes in genome, the extent of the change in the microbiome surpasses the extent of the change in the genome. So therefore, still we use the reference, human genome from NCBI to analyze the Indian genomes, but we cannot use the healthy microbiome. We cannot extrapolate the healthy microbiome to do case control studies in India. And this is one of the seminal studies that have showed it, that we consider bifidobacterium in the healthy gut of the Italians, but in the Hadza hunter-gatherers, they are very healthy, but they don't have an iota of bifidobacterium in them. They have Treponema, Prevotella, all these kinds of bacteria which we consider to be harmful in our healthy gut. So when I started my work in NIBMG, this was the thing that intrigued me, that the healthy human microbiome, although it performs essential functions, but how is it co-evolving with the humans? That is to look into the Indian populations, and we will look into the contributions of ethnicity, host biological factors, and the external parameters like food habits, geography, lifestyle, behaviors. How does they shape the reference microbiome in Indian population? And then how the host cell type in different organs. Now we can do single cell sequencing also. How the host cell type and the microbiome interacts in different organs of the human body for the sustenance of health. And when we understand this, we can then identify the perturbations in different organs that can lead to complex kind of diseases in humans with differential host genomic profiles. These are my students who are actually working day and night in our lab. So whatever I'm presenting here are mostly done by them. So why study the skin microbiome? Because the skin is the largest organ of the human body, and it encounters a continuous assault by the potentially pathogenic microorganisms. And the healthy skin microbiome actually keeps it healthy so that the opportunistic bacteria may not enter. And it exhibits a significant topological and temporal and inter-individual variations. And there are reports that the skin microbiome profile in the cesarean babies is associated with a risk of immune and metabolic disorders later in life. And skin microbiome is also important to understand atopic eczema, psoriasis, wound healing, and other kinds of things. So I don't know how to hide this one. Hide the video panel. Thank you. Then what can dysbiosis? I don't know. So yeah, let me continue. Thank you. So we started first with the how to identify the healthy human skin microbiome to investigate whether the inter-individual and temporal variations in the human skin microbiome can be significantly explained by the sebum and hydration levels in specific regions of the human face. It was funded by Unilever Bangalore, so face was important for them. So what we found is the sebum hydration levels we measured from the cheek and the forehead regions, because you see forehead is the more oily part than your cheek. And we found definite microbial abundances that are significantly, associating with changes in sebum and hydration. And we found differential predictive potential of the sebum and hydration levels. And we published that in scientific reports in 2016. That happens to be the first skin microbiome study on any healthy skin microbiome study on any Asian or Indian populations. And it is relevant because a lot of citations are happening now because skin microbiome is now being rejuvenated, restudied. What we found, interestingly, is that the European skin, we have a difference of the Streptococcus is more in our skin and Corynebacterium is significantly higher in their skin. So Corynebacterium is considered to be a healthy sweat detoxifying, nitrogen fixing bacteria in the skin. And Streptococcus, as we all know, are opportunistic pathogens. Although it was a 16 study, we couldn't do more at that time in 2016 up to the species. So with this, we started answering the question of atopic dermatitis. It's a dry skin disease, and we thought there are some evidences of filaggrin. Filaggrin is a gene. FLG is the gene, and filaggrin is the protein that is a skin barrier protein. And there are some loss of function mutations in Europe and Japan and China. There are many studies have been done, but no systematic study in India has been done. What are these filaggrin mutations and how do they predispose to AD? What we did here, we took AD cases and controls, and we took their lesional and non-lesional swabs. And here we did the shotgun whole genome sequencing. So we didn't restrict ourselves to 16S. We also took the human blood to do this filaggrin and later on other genes also. What we found that these are the loss of function mutations in filaggrin. Actually, there are so many. And some of them are found to be significant also in the different world populations. What we found that we did not find any of them in our cases and controls, although our sample size was less. So we had these genome Asia samples that were being sequenced in NIBMG, more than 500 healthy Indians, but we don't find these mutations. These are not like present or absent. These are present in the healthy population also in Europe and other parts of the world, only the frequency changes significantly. So we found that these are not all the 40 loss of function mutations are not present in either our sample size or in the entire Indian 500 whole genomes. So we started doing the microbiome, and here in the microbiome, we did this shotgun sequencing. The shotgun sequencing analysis pipeline we standardized in our lab, and these are having clade-specific markers for more than 17,000 microbial genomes. Gene families and pathways can also be identified. Pathway enrichment, although at the DNA level, can be done here. What we found that S. aureus is significantly higher and completely absent in the controls and significantly higher in the cases. And S. hominis is actually significantly higher in the controls. We moved more beyond that, and we found the gene family and pathway enrichment study where histidine and arginine metabolism pathway are significantly enriched in the cases, and these are all attributed to S. aureus MRSAs. As we know, and this is another pathway, Staphylococcus aureus infection pathway. These are the three important pathways we found enriched in the cases compared to controls. Now, we know that the S. aureus infection pathway has these gene families, and they confer resistance to antimicrobial peptides by inducing host inflammation. We also found that this histidine metabolism pathway, filaggrin is actually a histidine-rich protein. So this histidine metabolism pathway, it has a histidase and a urocanase. These enzymes are not present in the skin, and they can actually degrade the filaggrin protein. So even if we don't have the loss of function mutations, but this can be a probable mechanism by which the microbiome is actually mimicking that same phenotype. So we proposed this model, and this was an association study, and published this. What we are doing now is that we are now, with that shotgun samples that are done whole genomes, we are now identifying the bacteriophages, because phage therapy has now come up. As you can do phage therapy in combination with the beneficial bacteria. And these are some of the nitty-gritties of the analysis pipeline that we are using. We have found that the phages are more in the controls than in the cases. And there are some definite phages we are finding that why S. hominis is so less in the cases. We are finding that a lytic phage of S. hominis is actually more higher in the stressed condition in AD, and the S. aureus infection pathway that we are finding these genes can be attributed to these S. aureus specific some lysogenic phages that are integrated in their genomes. More work is needed to be done, and our host microbiome study is currently ongoing on larger number of samples. And also there is a whole battery of genes called EDC, epidermal differentiation complex, and filaggrin is only a single gene that we studied. We are now doing it. Now, moving on, we have also studied some chronic wounds. And chronic wounds, we are studying infected chronic diabetic foot ulcer. These are high-grade infectious diabetic foot ulcers. And we have started asking this question, why do most of the chronic diabetic foot ulcers eventually progress to non-healing state? And is there any microbiome associated with it? So we are doing a longitudinal study. And here we have taken wounds which are more than 2 months, and healing cases, non-healing cases, we are first taking their samples. And then after antibiotic treatment, when they are coming back, we are again taking their samples. This is a microbiome study design. So we are collecting many swabs and many wound biopsies for the biofilms and all. And we have still now reached the 100 samples, among which healing is 31, non-healing is 54, and we are having follow-up samples also. We are doing fish with the wound biopsies, and these are the confocal images of bacteria and the biofilm co-localization. We have done bacterial culture because we have to give back to the patient the antibiotic resistance report. What we are doing is we are actually identifying wound pathogens by microbial culture. In the total 153 isolates, we found more and more MRSA. So we have devised multiplex PCR to rapidly identify MRSA in the clinic. And this is based on a gene called a 16S-based gene to know the Staphylococcus genus, a NUC gene, methicillin-resistant gene, and a PVL gene, which will say that is it a community-acquired or hospital-acquired thing. And what we have done, we have taken 50-50 individuals from diabetic foot ulcer, diabetes without foot ulcer, and healthy individuals. And we have seen their nasal carriage and foot carriage and wound carriage also of MRSA. What we have seen is that the nasal carriage is 40%, to be significantly higher in the DFU compared to diabetic and non-diabetic healthy controls. And when we do a odds ratio correlation with the presence in the wound and presence in the nasal cavity, we find a 4-fold higher risk for wound MRSA infection in the non-healing diabetic foot ulcer patients. The odds ratio is 4.09. And the positive predictive potential is 664%. And most of these MRSA are MDR. We have just recently published this in BMC, that the nasal MRSA carriage is a risk factor for the antibiotic resistance in diabetic foot ulcers. During the COVID time, we are also doing sewage surveillance. I'm the nodal officer from the state of West Bengal. But here we have done some microbiome study on Delta and Omicron patients compared to healthy controls. What we have seen is that the healthy controls were abundant in the commensals, but when we compare the Delta and Omicron, the Omicron is more similar to the commensals, but the Delta is having more and more Klebsiella pneumonia, Acinetobacter baumannii, and you can see this, this principal component, Delta is completely different, and Omicron and healthy has this overlap. We don't know why. We have found out a 8 to 9 bacteria trend test analysis, and we have seen they have a gradient from control to Omicron to Delta. And by random forest machine level analysis, we could identify with these microbiome 90% accurately who is a Delta, who is a Omicron, and who is healthy. We have published this recently in Spectrum. And our lab is doing atopic dermatitis, psoriasis also we have 300 samples now for psoriasis cases and controls. We are also doing another arm is preterm birth and childbirth failure. These are the 5 publications from our lab in recent times. And one of my students is also working on childbirth failure. And here we know that that is very important now because in the global hunger index, India is going down. And one of the reason is childbirth failure, stunting, bursting, and underweight. I will not say more about it. You have to go to this poster. My student is sitting in the audience. This poster, we have 3 months and 6 months stool samples collected. It's the WINGS cohort from CHRD SAS. We are using this systematically to understand both the intervention arm and the control arm. So what is the change in the microbiome? How is it associated with childbirth failure? NIBMG has core laboratory facilities, a zebrafish facility, the largest genomic facility in any government or organization in India. And correlating to it is the largest computational also. We have a bioplex facility now, a proteomics facility has come up, flow cytometry, confocal, and we have now a human microbiome facility where we do, other than the association studies, we can now do the anaerobic culture and other functional co-culture assays. We are expanding. We have collaborated with IMTech Chandigarh to do an oral cancer tissue resident microbiome that is published in Spectrum. We are collaborating with THISTI and CHRD SAS in Haryana area for this Garbhini cohort and also the child cohort. LVPI Hyderabad has recently reached out to us and collaborated for a nationwide diabetic retinopathy gut microbiome, more than 4,000 samples, a shotgun sequencing study. Unilever Bangalore is our friend for the last 8 to 10 years, funding skin microbiome because that's not funded by any government organizations. It's not a public health problem per se. We are collecting samples for IPGMER, CNMC, MR Bangur Medical College in Kolkata. And with the northeastern states, we are doing the sewage surveillance day in and day out. These are some of the co-authorship that we have done with these collaborators all over India. We have organized in November 2019 an India EMBO Symposium on Human Microbiome Resistance and Disease, and I look forward to someday hosting this probiotic symposium in NIBMG. And these are some of the glimpses. Some more than 15 European microbiome people came. We thought to continue this, but we could not because the COVID came and everything got stalled. These are my two students who are doing all the work, and thank you all for the patience. Jai Hind.
Thank you, Dr. Savik Mukherjee. You went with the theme, you went beyond the intestine, and you talked a lot about the skin microbiome. So we've moved from the gut to the skin microbiome. Now, I would like to invite all the speakers, Professor Ahuja, Professor Chandi, and Dr. Savik, please, Professor Savik, please set up on the dais, and the house is open to questions. Yes, Dr. Sarath.
Thank you. Dr. Savik, your presentation was wonderful because it appeared to have a lot of clinical applicability, and there's a huge potential for clinical correlation. Now, my question to you is, when you look at atopic dermatitis as an entity, being a pediatric gastroenterologist, I'm specifically interested because the kind of immune activation mechanisms in infancy which cause atopic dermatitis. For example, milk protein allergy. One of the presentations is atopic dermatitis, and it's non-Ig mediated. But the kind of activation pathways which occur at that age may largely depend or almost predominantly be dependent on the gut microbiome, because that's where the allergy started first. The same atopic dermatitis, when it happens in a much older child or a teenager, let's say because of a cosmetic reason or some, are the immune mechanisms fairly different? Because, or is it directly triggered by the skin microbiome? What's the difference between these two?
Yeah, it's a very relevant question. We, since this is funded by Unilever, we couldn't include children in them. They had an ethical problem in their company because since they don't work on childcare products, so they couldn't allow us to include children. But the clinicians kept on saying, if you are not doing children, why are you doing this? So this is a thing that we had to address. So we are now starting some work from some intramodal funding from NIBMG on the children to address this question. And the second thing is, yes, there are environmental stimuli, as you say, some exposure to some chemicals or something in the adults that can create AD. So for choosing atopic dermatitis, we had depended on this Hanifin and Rajka criteria. And this criteria has a long list of questions, starting from, have you got this in childhood or have you now got it in adulthood? And if you know for sure that atopic dermatitis, how to diagnose that is varying from clinician to clinician. So since we collected from 2-3 hospitals, we had all these clinicians come together. They agree on a set of questions and the answers, how they will judge the outcome. So that effort we have taken, but yes, we have measured the IgE also. I have not shown here, but child we are now doing.
We're actually taking this further. Skin is the biggest immunologic organ in our body. It has dendritic cell, local dendritic cell, mast cells, huge number of things which it has. And the dermatology has really moved tremendously forward. Now there are specific monoclonal based therapies in psoriasis and all these things. So this we didn't talk about immunome today.
Yeah.
But when we are doing multi-omics, the immunome becomes a very strong part if we are talking of inflammation, if we are talking of host-pathogen relationship. So this will be very like one of the psoriasis trials which we are doing. The major regulator was IL-10, and this has to be assayed in a United States laboratory.
Yes.
I have one comment for you. That a comment is that there is a ratio of Corynebacterium diphtheria, Staphylococcus aureus, and Streptococcus pyogenes on the skin. There are other Streptococci and other which are non-pathogenic. this ratio need to be studied.
Okay.
Because and these are not homogeneous, but like the Staphylococcus aureus, I don't know Rama Chaudhary is sitting here or not. There's a Baird-Parker classification for that. There are coagulase positive, coagulase negative. So in the same way, staph pyogenes in the skin and the throat are totally different population. Throat one causes rheumatoid heart disease, the skin one causes impetigo and glomerulonephritis. So you need to really collaborate with a microbiologist or a nephrologist or somebody to this.
Sure.
So Dr. John, I have a small comment for you that the VSL3 which you are using is not a VSL3 because the strains are not the same. The company fell out. The person who had the strains is known as the Simon, and now it is known as Professor Simon strains, and it is being manufactured in another name in India. So all those VSL36 strains which were present in VSL3 are not the same. But I was very much impressed by Dr. Savio. I never saw a gastroenterologist going in such a depth in this kind of a study, Dr. Ahuja. I have one comment to make. Today, Dr. Mathan's name was taken by Dr. Ramakrishna, Dr. Chandi, and he was one of the earliest person to study the gut microbiota in India. There was a guy from Gorbak. His name was, he was studying in the tropical medicine. So they found that the upper gut flora in Indians is very, very heavy, which is not there in the temperate zone, Europe and Japan and other places. So when we are giving fecal microbiota from the upper gut, the fecal transplantation from the upper gut, we are populating duodenum. I don't know whether we are doing harm or whether we are doing if it could go a little lower, it will perhaps be better. And we have Dr. This is the last comment. We have Dr. A pathologist in our place in PGI. He wanted to find a normal person. So he took about 1,000 rectal biopsies from so-called normal person. Every rectal biopsy was very, very inflamed. So in my definition, the normal person will be that if we do metabolome signatures, and I have a state-of-art metabolome laboratory. Actually, Sobhi, whatever you showed there, I have it in a private hospital. So the metabolome signature will be very nice. And Sobhik, we have skin organoids. Like he has the gastric, the skin organoids could be one of the places to study. Thank you.
Yeah, I think there are two.
I have a brief question to Professor Ahuja. A very, very nice presentation with it. So you talked about urolithin. I was a little curious about that. The name suggests that it might cause kidney stones. Do you have any comment?
So this question was asked to me also by Dr. Narendra Nathan and Dr. Katie Shinoy. So at that time, I didn't know. I looked back into it, but I could not find any such connection as the name suggests. It is basically elagilic acid, which is an extract of elagilic acid, which is found in pomegranates, raspberries, and all that.
Because doing something should not, there's something called the law of unintended consequences.
So I just had a question. Well, excellent presentations. Dr. Vinita Ahuja, that's very nice. We know the power of FMT today in treatment of so many diseases. But just wondering, what is the staying power of FMT? Do you need to do a repeat? Because all these microbes are transient colonizers.
That's an excellent question. It depends upon the disease. Right now, FMT has been globally approved for only one indication, that is recurrent or refractory Clostridium difficile infection. Now, that is an acute infection. So because it's an acute infection, there's a acute shift in the microbiome niche out there. So the moment you repopulate that microbiome niche, it results in a change in the course of the disease. For chronic diseases like inflammatory bowel disease, there seems to be a chronic change which is occurring out there. It's like a perpetual forest fire out there. So just a few pails of water is not going to quench that fire at all. So obviously out here, what has been seen is that if you give FMT, it may last for 8 weeks or 12 weeks. Again, subsequently, you'll have to do another microbiome manipulation therapy. It is what we have been seeing with infliximab, which is a TNF inhibitor. You give it, the response is there, let's say for 8 weeks. When the drug levels go down, you have to give another injection.
I thought this will be a panel question, so you have already asked this question. Regarding the C. diff, what he's saying is very correct, but the problem comes when it is relapse or it is a recurrent C. diff infection, which is quite common in some cases, and some of the pediatric cases we have seen. So there is an indication. There we can give repeat FMT. And it has been shown that if you give just once you give FMT, it is giving 84% success rate as per the genomic analysis done in 2023-24. And this has been just recently published. But if you give repeated ones, there is almost 92% success rate. And we do need to give because in many countries, though this problem is not so much in India, but in many countries, they have found that relapse is a big problem. And even fidoximicin, which is now the newer drug for C. diff, doesn't work effectively. So FMT is the only choice left in those cases. You have to repeat in that cases. Yes.
We have one more question from the.
Just one second.
Yeah.
The data which we presented yesterday on the FMT, I don't know whether you saw that or not. We followed up the microbiome, the things which we had given, strains which we had given, how long do they stay? It was not a very long follow-up, but we were able to show how long they stay. So that's one of the major components we need to put up in our analysis.
Yes.
So around what we've seen is 50% till 6 months or something like that, engraftment.
Yeah, your question?
So I have a question to Savik. You showed very high MRSA carriage in the diabetic foot ulcer patients. Seeing that this is a probiotic conference, I was just wondering, would you think that nasal application of probiotics might be preventive in these patients?
So interestingly, one faculty from Ohio State University visited us last week, and he was giving examples of when they have any surgery. Now, for the diabetic patients, they apply anti-MRSA things, ointments in the nose and skin. So yes, so I just could correlate that thing with the findings, and I told him in diabetic foot ulcers, we are finding that. And yeah, so I don't know about probiotics, but anti-MRSA creams and all, if they are available, but that will create antibiotic resistance again.
So I would like to add here, there have been bacteriophage applications locally in nasal, which are specific against MRSA, and they are found to be effective. I don't know about any probiotic which is specifically used for that, but yes, the bacteriophage application is available.
Dr. Vineet, I have a question. I was really interested in the term you used, anti-inflammatory diet. You did show a slide, but I wanted to understand, is it based on certain components which are there in the diet, or is it only like what I could see was gluten and lactose? Yeah.
So it is based on all those components. They have experimental or mice data available or cell line data available that they promote or decrease inflammation depending upon the component. Second philosophy behind that diet was to increase the aryl hydrocarbon rich ligand diet, like give more of cabbage, cauliflower, radish, and etc. These things. So it was based on these two things. And so for IBD, you have a lot of exclusion diets which are in vogue, I would say, like anti-inflammatory diet, or we have the Mediterranean diet, which is used. Then you have the Crohn's disease exclusion diet, which is also largely based on these principles. So essentially, most of these diets are more amount of fresh fruits, vegetables, processed food, ultra processed food, packaged food goes away. To avoid red meat, fish is fine, egg is fine, and gluten is something which probably promotes inflammation, so most people want to avoid it. Milk, probably because of lactose, as such people want to avoid it. Curd and buttermilk.
So it means it is based on
It is,
identified anti-inflammatory components in food,
yes,
which has been experimentally
Yes,
done.
yes.
I would be very interested to know more about it.
Yeah.
One more thing which I observed in your presentation was that you spoke about FMT in weight loss. I was quite intrigued.
Obesity, obesity.
Is it?
No, these are like what slide I showed you that had a combination of case studies, case reports, case series, randomized control trials, cohort studies. So depending upon the disease. So in weight loss or obesity, we do not have any randomized control trials. These are just anecdotal case reports.
Okay.
That was just to show the extent of the expanse of application of FMT, that it is not related only to GI diseases, but its application is beyond GI diseases.
Okay. And one last question to Dr. Souvik. Dr. Souvik, have you done any food-based intervention in relation to skin
No.
and the skin microbiome?
No, we haven't yet done any interventional study. Mostly these are observational
Just
studies.
observation.
Yeah, but we are now going to the, so like in the plenary talk, he said stage 1, stage 2. We are into stage 2 now. Now we are going to stage 3. Before going to stage 3, we are doing the validation in this in vitro models. As Professor Ganguly told, organoids and all, we are doing co-culture and these types of things. Once we know the mechanism, then I think we can go for the stage 3.
Thank you. Yeah.
May I ask one question from
we'll,
online?
yeah, so we'll just.
Mr. Chairman, can I ask a question?
Okay, yes, please go ahead.
Yes, I am A.K. Srivastava, Vice Chancellor of Mathura Veterinary University. My one question may not be directly related to the clinical side, but when we talk about gut microbiota, do we have sufficient information in literature that how gut microbiota interfere or interact with inflammatory mediators, maybe bradykinins, maybe histamine, or maybe cytokinins or prostaglandins? How with these inflammatory mediators, maybe any disease which are causing the inflammation, but as far as inflammatory mediators are concerned, how they interact?
So Professor Srivastava is basically addressing the mechanism of action.
Yeah, so there is plenty of data available, studies available, which have shown that how gut microbiome primes the immune system. In fact, it educates the immune system, and it can drive the immune system at various points of life in infants, in early adult, adolescent, elderly age. So there is clear-cut data available about the host microbiome interaction, of which immunological mechanisms and microbiome and metabolites, the data is clearly available.
Yeah, thank you. Okay, we have two questions.
Thank you very much.
One is addressed to Professor Chandy. The question is from Professor Prakash Halami. He's wanting to know Bifidobacterium, which was supposed to be the so-called good probiotic, suddenly seems to be cropping up in high levels in disease. So is there a sudden shift which is there, or you can highlight anything you can highlight?
Ajay, this has been looked at in great detail in several studies, and the impression is that this is the body trying to respond to the need. So the reason why you find bifidobacteria in large numbers is
so
because
it's a cause and effect, basically.
It is a
not
response.
Cause, it's the effect.
That's right.
All right, all right. And for you, Professor Vineet, the question was, one was on urolithin and the other was on potassium.
We talked about coconut water. If potassium was so important, do you think addition of pot chlor to the prescription would help?
So pot chlor, number one, it would cause a lot of gastritis. If somebody has to take 15 to 30 ml of pot chlor every day, nobody will be able to take it. Coconut water, besides being having more of potassium, it obviously has a lot of health enhancing properties. It is a part of a good diet, right? So I would say that yes, you can give orange juice that would increase potassium. So you can work on it and show proof of concept. We picked up coconut water because we thought that if we want to give something which is palatable, which is healthy, and at the same time it leads to, for the patient would be happy to take it. So we zeroed down on coconut water for those.
Pomegranate also has potassium.
So pomegranate has
that also.
Pomegranate has urolithin also. So that will be a great combination of potassium and urolithin.
Yeah, maybe that would be better.
That would be the next.
I think we will close this session now. Thank you so much, Professor Ahuja, Professor Chandi, Professor Banerjee. Thank you so much for an excellent deliberation. A big round of applause for our three speakers.
I think I must add that all the speakers were within time and they were absolutely on dot. So congratulations. So thank you so much, everyone. And now it's time to felicitate our keynote speakers, requesting chairpersons to felicitate the keynote speakers. Yes. So let's take a photo with the chairpersons and the speakers. And now I would like to welcome Dr. Jyoti Tamang and Professor B.S. Ramakrishna to felicitate our chairpersons. Yeah. So thank you so much. And I request for a group photo, please. So please join for a group photo. Yeah. So thank you so much, everyone. All right, so ladies and gentlemen, now we'll take a break for a tea. So enjoy your tea and come back by 12:15. I'm sorry, by 1 o'clock, please. So before that, there are some reminders. All the delegates who has registered will get the e-certificates on your email IDs. And the top 20 young investigators can collect your participation certificates from the registration counter outside. So make sure that everyone is reporting back for the next sessions by 1 o'clock. Okay. So also, please submit your survey forms to the registration desk.
So I request everyone to please submit your survey forms which you have received at the registration counter.
Yes, sir. No, no, Akshay, Aditi, go ahead. Three of them. And the fourth one is also there. I just need to ask that
All right, so I think everyone is back after the tea break. So we'll continue our sessions, and this will be young investigators' presentations. I would like to welcome Professor Jyoti Tamang and Dr. Sharad Gopalan onto the dais. So both of them will be chairing for young investigator presentations.
Should we start?
Hello. No, no. Immediately. Those who are outside, please.
Yeah, they should be here.
Ladies and gentlemen, for your kind attention, if any of our young investigators are outside the venue, outside the hall, please come inside and join for the session, because this is something really important, and we'll definitely have some thought-provoking questions or session. So requesting all our young investigators to join together inside the hall.
Start. Yes, start. Good afternoon, dear colleagues. This session is actually a very, very important session, because if you actually look at the future of documentary evidence when it comes to scientific literature and any new work going on in the field to update hard evidence-based information, it all depends on active research. And the future of these active research initiatives are to be taken by a younger generation. It is our youngsters who will lead the way. And this Young Investigator Award is a very, very important session from that standpoint, because these are the youngsters who have done original pioneering work, which may have a huge applicability and may influence not only certain clinical decisions with applicability, but also in some situations, maybe policy changes. So this becomes an important session, and we really are happy that we have had a number of young investigators coming forward to present their abstracts in this symposium. And they were really high quality abstracts. I now request Dr. Jyoti Tamang to tell us more about the Young Investigator Awards, how these awards are processed, how one went about it. Please go ahead, Doctor.
Namaskar. One of the very important flagship program of our foundation is to encourage the young researchers or young investigators working in the probiotics and the related fields. And every year, along with our international symposium, we also organize the poster awards, posters for the young investigators. So the process is that we invite the posters in our website and our contact to all the university institutes by our secretariat, mostly by Dr. Nirja. Then after that, we get a nominations, we get a applications. Then they do the screening. After that, they evaluate the applications. The poster, the abstracts are being evaluated by the members of the governing body members of our foundations. But the condition is that the students of the member will not be evaluated by, suppose if I have a student, I cannot evaluate my own students. So that is the thing. Then after that, we select the best investigators, and they are asked to present here, come, we'll provide their travel and the other logistics. Then they are asked to display the posters, and there is a jury. The jury is not from our GUT Foundation members, as an international independent jury. So they select first, second, third every year. So this year we have received 51 applications. Out of 51 applications, 20 have been shortlisted, they have been selected. You are here, all 20. You made your presentations, you made your the poster display, and our international jury has already evaluated, and the marks is with me. So with my friend Dr. Gopalan and me, we have the list. I will go with the third list, third name, third prize, then second, and the first. So when I announce the name, you can just stay there. Then after that, we will invite one by one for your presentations, only 10 minutes presentation, exactly 10 minutes presentation, and there will be no question answer, and you will be awarded. The third prize goes to Aditi Goel of CFTRI, myself. Please stand up. Where is Aditi? Okay. Second prize goes to Akashi Gaike of SPP University. Now we have first prize, but there are two equal marks. So I'll go by the alphabetic name. First, Ayushi Purohit of the Translational Health Science and Technology Institute. Then first, another first is Manusurya Palapu. Okay, now I invite Ayushi Purohit to make a presentation, 10 minutes exactly. And you'll be given the warning at 8 minutes.
So good afternoon, one and all. So my abstract was entitled as a gut signature linked with increased ethanol production and liver inflammation contribute to non-alcoholic fatty liver disease in Indian patients. Yesterday, I had a talk about this topic, but today I'll go in more crisp details. So non-alcoholic fatty liver disease terms refers to a range of spectrum of disease with excess accumulation of fat in the liver. If the fat in the liver is more than 5%, it is known as non-alcoholic fatty liver. The spectrum of pathology start with steatosis through inflammation to fibrosis and ultimately leading to hepatocellular carcinoma. This silent epidemic is on horizons because it has no therapeutic to the disease. So there is a bidirectional link between the microbiome and the liver that is known as the gut-liver axis. The portal vein carries all the microbes and the microbial metabolites and transmits to liver, which is one of the critical risk factor for the disease. This disease is on horizons because there is no therapeutic till date for NEFL. You can just reverse by consuming 800 units of vitamin E, weight loss diet, regular aerobic exercise, eliminate alcohol. If diabetic, tight control of glucose level, lose 7 to 10% of the total body weight. So the key risk factors that associates from transferring to NEFL to NASH, which is one of the severe form of the disease, are genetics, bile acid, gut dysbiosis, lipotoxicity, and environmental factors. So I have worked concurrently on gut dysbiosis part. So the sampling was done in the NCR region of Delhi. We collected the sample from 3 cohorts: healthy, obese, and NASH. Healthy were 22, obese was 17, and biopsy proven NASH were 10 that were done with Dr. Shalimar from AIMS. So this is the characteristic of the people. We took the diet that is the vegetarian, vegetarian, and the non-veg diet, and we took the biopsy approvals from the AIMS. So when we calculated the core OTUs, we found 12 OTUs were present in all 3 cohorts, that is healthy, obese, and NASH. Based on the median value, we calculated and we found Colensella aerofaciens was one of the taxa that was increased rapidly from healthy to NASH. As obesity is directly linked with NEFL, we tried to illustrate the genomic repertoires of Colensella aerofaciens in Indian population. We took biopsy-proven NASH, isolated this Colensella, screened it with the specific primers, and did the whole genome sequencing at THSTI at MySeq platform. The complete genome had 2.3 MB in length, 60.1 GC content. The genome had enriched itself in the virulent 29 to 30 more genes that were virulent, and more than 258 genes were involved in carbohydrate, fatty acid, and lipid biosynthesis. So there was a remarkable feature from our study that we found a SNP that is present in the NASH isolates that was mutated and was leading to high alcohol production in the gut. So if these people are the people that were not having alcohol, but the bacteria was making alcohol in the gut. So this is the amino acid sequence that we have sequenced. You can see the mutation. This reference is from the healthy person, and this is the mutated gene at the SNP level of all the isolates that were isolated from the NASH individual. We started with the animal study. We took C57BL6J male mice, 8 to 12 weeks old. This was the schematic of the work that we have done. We have taken the normal chow diet and the CDHF. We have given 2 doses of the bacteria subsequently after the weeks at day 28 and day 35 and given through the oral gavage. We took the body weight, the serum samples, and the liver for the downstream processing. So these are the things that we have measured. We have measured the circulatory ethanol levels, the fibrotic parameters, inflammatory parameters, lipogenesis and lipolytic parameters, hydroproxylene, triglyceride, and histopathology analysis was done. So this is the triglyceride analysis. You can see the mice that were fed with CDHF and Colensella showed a high increase in the triglyceride level. The macrosteosis you can see in the cohort of CDHF plus Colensella showed macrosteosis and microsteosis. It means that is what inducing the inflammation in the liver. This is done by GCMS. We took the mice blood and calculated the ethanol concentration. So you can see the mice that was fed with normal chow also, but if given Colensella aerofaciens, was increasing the ethanol production in the mice liver. So the hydroproxylin and the triglyceride ratios were also increasing with CDHF plus Colensella aerofaciens. These were all the inflammatory markers and the fibrotic markers like NF-kappa beta, TNF-alpha, IL-6. IL-10 is an anti-inflammatory marker. You can see in the cohort with CDHF plus Colensella, it was tremendously decreasing. Alpha SMA, Col1, alpha, TGF beta fibrotic markers. We also seen the fat uptake and the fat releasing genes like PPR alpha and fat P5. So you can see that fat P5 and PPR gamma was increasing with Colensella aerofaciens. So the conclusion of the study is we study the gut microbiome of NEFL, obese, and healthy subjects. We observed a higher abundance of Colensella aerofaciens in the obese and NASH patients. Cholinceriofusion contributes to increase in the level of systemic ethanol in the mice. Increased ethanol, hydroproxylene, and triglyceride induce hepatic inflammation. The insight of this current finding is the first study to correlate the genomic signatures of cholinceriofusions and association with NAFLD and disease progression. So there are several limitations of the study. In this study, host bacterial interactions were not explored. The study are primarily focused on the bacterial taxa, as we know that microbiome is coming in limelight, which based on the fungal component has not been elucidated. This has been done only on 10 samples, so it is having a limited sample size. The cross-kingdom interactions have also not been done, so this study has to be validated in a larger cohort. This is the graphical abstract. We have seen that low level of Collinsella aerophaciens lead to low ethanol production, which can lead to less inflammation, but high abundance of Collinsella can lead to high ethanol and high inflammation. We have done the NGS, GCMS, qPCR, and microtome. So if you want to go in detail, you can go through the article that has been recently published, Collinsella aerophaciens linked with increased ethanol production and liver inflammation contribute to pathophysiology NAFLD in eye science. So the minds between this study are Dr. Bhavatosh from THSTI and Dr. Sanjay Banerjee from NIPER Guwahati, who are my supervisor in this study. So thank you.
Thank you, Dr. Ayushi.
Thank you.
Thank you, Dr. Thank you, Dr. Ayushi. We now move to the second speaker, who's also got the first prize. Manisurya Palepu from the National Institute of Pharmaceutical Education and Research, NIPER. Over to you.
Good afternoon, everyone. Good afternoon, everyone. My name is Manisurya Kumar Palepu. I am from NIPER, Hyderabad, and I'm working under Dr. Manoj P. Dandekar. This is my thesis work. The title is Short Chain Fatty Acid Supplementation Rescues Anxiety and Depression-like Phenotypes Generated by Fecal Engraftment of Treatment-Resistant Depression Rats. Treatment-resistant depression. This is the basic pathophysiology of the depression. In this, there is an alteration in the HPA axis and the tryptophan metabolism, and there is an increase in the neuronal inflammation and neuronal death, and the neurotransmitters get dysregulated. Rational. In the major depressive disorders, we have given this, like when we give the antidepressants, some are responders or some are non-responders. So here we are elucidating how the gut microbiome composition differs between the responders and the non-responders. That may be the novel therapeutic treatment. This is the methodology. So rats were subjected to the chronic unpredictable mild stress for the 6 weeks period of time, and adrenocorticotropic hormone for the 2 weeks period of time. 2 week period of time, and we have given the short chain fatty acid treatment for a 1 week period of time, and we did the fecal microbiota transplantation from the control rats to the healthy recipient rats, which was named as control FMT rats, and we did the fecal microbiota transplantation to the from the TRD to the healthy recipient rats, which are the TRD FMT rats. These are the methods and instruments we have used in this study. Coming to the results and discussion. Here you can see we performed the behavioral parameters, sucrose preference test, forced swim test, and open field test. And in these, the depressive like behaviors were increased in the TRD and the TRD FMT rats significantly. And after the administration of short chain fatty acids for a 1 week period of time, it got reduced. And coming to the anxiety-like behavior, we did elevator plus maze test. And here also you can see the percentage entries in the open arm and percentage time spent in the open arm are decreased in the TRD and the TRD FMT rats. And it got reversed when we given the short chain fatty acid treatment for a 1 week period of time. And coming to the FFAR receptors, free fatty acid receptors, we did the RT-PCR for the FFER2 and FFER3 receptors in the frontal cortex. And you can see there is an increase in expression of the FFER2 and FFER3 in the after giving the short chain fatty acids as a treatment. And we quantified the corticosteroid and the BDNF. In the plasma, the corticosteroid levels are increased in the TRD and the TRD FMT rats, and after giving the short chain fatty acids, the levels got reduced. Next, we quantified the BDNF levels in the frontal cortex. You can see in the TRD, the levels got reduced, and after giving the short chain fatty acids, the levels are increased, restored. We also did ELISA for the pro-inflammatory markers for the TNF alpha and IL-6 in the frontal cortex and the hippocampus. So you can see the TNF alpha levels are increased in the TRD rats and TRD FMT rats, and in the frontal cortex we have not observed any significant different changes in the TNF alpha levels, but we observed significant changes in the TRD FMT rats. And coming to the interleukin-6, in the normal group, we have not observed any changes. But in the FMT groups, after giving the short chain fatty acids, the level got reduced. And the interleukin-6 in the hippocampus, there is no significant changes in across the groups. And we did RT-PCR for the C-reactive protein. The C-reactive protein increased in the TRD, and after giving the short chain fatty acid treatment, it got reduced. And we also estimated the BDNF and double cortin levels, which are neurogenic markers, and the levels got reduced in the TRD and TRD FMT rats. Ocridin, we also did the mRNA expression of the ocridin. This in the ocridin levels got reduced in the TRD and the TRD FMT rats, but we have not observed any changes in the FMT groups. Coming to the neurotransmitters, we quantified the neurotransmitters in the LCMS triple quadrupole. You can see the levels of serotonin, dopamine, GABA, and norepinephrine are reduced in the TRD and TRD FMT rats, and after giving the short chain fatty acids, the levels were restored. And the SCFS reversed the altered tryptophan metabolites. The tryptophan and the cyanuric acid are they are having the protective nature. These levels got reduced in the TRD and the TRD FMT rats. And after the short chain fatty acid treatment, it got reversed. And the cyanuric and the quinolic acid are the neurotoxic compounds in the brain. These levels are increased in the TRD and TRD FMT rats. We performed the immunohistochemistry of corticotropin releasing factor in neurons in the paraventricular nucleus region of the hypothalamus. In the TRD and the TRD FMT rats, you can see the CRF neurons, but after giving the short chain fatty acids, in the TRD1 it got not reversed, but in the TRD FMT rats it got reversed significantly. And we performed the immunohistochemistry for the jonocridin protein in colon, and you can see the B and E, TRD and TRD FMT rats, the expression levels of jonocridin got reduced, and after giving the short chain fatty acids, the levels are increased significantly. And we performed the short chain fatty acid concentration analysis through the NMR in the plasma and fecal samples. And you can see the levels of acetate, propionate, and butyrate were reduced in the TRD. There is no significant changes in the levels of acetate in the plasma, but there is significant changes in the levels of propionate and butyrate after giving the SCFS. And we performed the SCFS concentration in the fecal samples also. And here you can see there's a reduction in the acetate, propionate, butyrate in the TRD and TRD FMT rats. We did a we did a HNE staining and Elsham blue staining. In the TRD and the TRD FMT rats, the Villi to crypt ratio got reduced, and you can see the neutrophil infiltration in the TRD. And after giving the short chain fatty acids, it got reversed. And in the Elsham blue staining, the number of goblet cells are reduced in the TRD and TRD FMT rats. And after the treatment with SCFS, the number of goblet cells got increased. And on top of that, we performed the 16S metagenomics. Here you can see the some different changes are there in the relative abundance in the phylum level and the family level. And here we observed changes in the genus of Faecalibacterium, Subdoligranulum, Solobacterium, and Peptococcus in the TRD as compared to the control rats. A conclusion is rats which are exposed to chronic unpredictable mild stress and ACT has showed the depression and anxiety-like behavior. And rats induces the changes in the gut microbiota composition. The short chain fatty acids reduce the levels of TNF alpha and IL-6 in the frontal cortex and hippocampus. And there is an increase in the CRF expression in the paraventricular nucleus of the region of the brain. And the short chain fatty acids normalizes the levels of neurotransmitters like serotonin, GABA, norepinephrine, dopamine in the frontal cortex, and tryptophan metabolites in the plasma. And it got the levels of neurogenic markers like BDNF and double are reduced in the hippocampus. The keynotes. Short chain fatty acids, they may have the beneficial effects by the modulation of tryptophan metabolism, inflammation, neurotransmitters, and neurotrophic factors, and the brain access through that. So robust clinical trials are required to validate the potential therapeutic effects of the short chain fatty acids in the TRD. We are thankful to the NIPER Hyderabad Department of Pharmaceuticals, Ministry of Chemicals and Fertilizers, and IBRO International Brain Research Organization for their support in conducting the present study. And this work got accepted, in a 2 weeks back in the ESS Chemical Neuroscience, and that one is the cover page in the ESS Chemical Neuroscience. That also got accepted. Thank you.
Congratulations, Manasuriya, that you got this accepted. It's a good impact factor. We now move to the next presentation for the second prize, Akshay Gaike from the National Center for Cell Science and Department of Zoology, SVPU. Akshay, please come.
Good afternoon, everyone. Myself, Dr. Akshay Gaike. I am the title of my abstract is title of my study is effect of long-term oral glutathione supplementation on gut microbiome of type 2 diabetic individuals. I work with Dr. Yogesh Sauche from NCCS and Professor Saroj Gaskadbi from SPPU Pune. So diabetes is a complex multi-system disorder and its prevalence is increasing worldwide. It's around 2 to 20% of the global population is affected due to diabetes disease. In India, it constitutes around 19.3% of total number of diabetes in the world, and approximately 0.6 million deaths have occurred till date due to the diabetic condition. And it also puts the high burden on healthcare system in India due to these diabetic conditions. So there are major factors which are affecting to the diabetes development of the diabetes, like inflammation, altered lipid metabolism, oxidative stress, which is resulting due to the wrong dietary habits and sedentary lifestyle. Recently, the gut microbiome has been identified as a major contributor in the development of diabetic and its complications. Previously, we have published this data where we found that the abundance of the in the observed diversity, alpha diversity, we found that the alpha diversity was decreased significantly in the newly diagnosed diabetic individuals. However, after the anti-diabetic treatment, the diversity is slightly increased and restored to the normal one. And based on the beta diversity analysis, where we found that the inter-individual variation is more in diabetic individuals compared to the pre-diabetic and healthy one. However, pre-diabetic is slightly overlapping with the diabetic individuals. So, based on this, we have proposed the study where it is known that previously is known that oxidative stress is involved in the development of T2D. Among various parameters of oxidative stress examined previously from our lab, we found that the glutathione was the best covariate for the glucose recovery. It is also published in one of the journal where we have conducted a trial for around 250 individuals where we found that the GSS lowers the glycemic condition in the type 2 diabetic individuals. So here in this study, there are a control as well. This is a case control study where control individuals, which are known as diabetic alpha for the day 0, and control individuals after 6 months are known as diabetic gamma. Then case individuals were like diabetic plus they have given the GSS supplementation of around 500 mg per day, were termed as the diabetic glutathione alpha, and the group after 6 months is known as diabetic glutathione gamma, like DG gamma. So stool and blood samples were collected. Blood was collected by trained phlebotomists from the Golwilkar Diagnostics Lab in the Pune, and stool sample was collected for the microbiome analysis, where we have for serum biomarkers, we have analyzed the fasting glucose, HbA1c, fasting insulin, GSH and GSSHG, and 8-OHDG, which is oxidative DNA damage marker. And from stool sample, we have extracted the microbial DNA and went for the 16S RRN gene for the next generation sequencing using Illumina platform, and analyzed using the Data2 platform for the microbiome profiling. In serum biomarkers, we found that after glutathione supplementation, in DG gamma group, after 6 months, the fasting glucose and HbA1c were significantly decreased after long-term supplementation. However, the GSH was increased significantly after 6 months. It suggests that the body stores of GSH was maintained in the DG gamma group after 6 months of treatment, where 8-OHDG was decreased significantly after the GSS supplementation. It suggests that the DNA damage marker is decreased drastically after the treatment, leading to recover the oxidative stress in the body, reduce the oxidative stress in the body. However, HOMA beta was also increased in treatment group compared to the control. It suggests that the beta cell function of the individuals were recovered compared to the normal diabetic individuals. Then we moved to the to analyze the microbiome of the these individuals, and we found that the phylogenetic diversity was not changed significantly in both the groups. Then we moved to check the Simpson dominance for the dominant taxa which are present in the overall gut microbiome, and we found that the dominance taxa were increased significantly in DG gamma group after the oral supplementation. However, the intra-individual variation in the beta diversity was seen in this group. You can see that. Then we moved to analyze the overall microbiome profile at the phylum level, and found that among the top phyla, we found that Proteobacteria was found to be significantly decreased after the treatment, 6 months treatment. Then the genus level, the general level microbiome analysis was done, and we found that the genus Bacteroides megasphaera was found to be significantly abundant with increasing trend, while genus Escherichia and Shigella exhibited the decreasing trend in subjects with the glutathione supplementation. So previously we found that the body stores of glutathione was increased in the individuals after treatment. Then we have decided to check the effect of this glutathione supplementation at the intra-individual level, and we found that in DG group, where we are given the supplementation, the decrease in the Escherichia ASVs, like amplicon sequence variant, was found to be decreases in each individuals around almost all the individuals in DG group. And however, the beneficial microbes like ASV2 for the megasphaera were found to be increasing in all the diabetic individuals after the 6 months treatment. So the final conclusion is, this is the first report demonstrating the shift in the gut microbiome community structure and function from healthy to diabetic state in the Indian population. Our findings suggest that the gut microbiome diversity significantly decreased in new DMs, but not in pre-DMs, and was restored in known DMs on anti-diabetic treatment, or to some extent. The glutathione supplementation given to each diabetic individual, which lead to increase in the body stores of glutathione and improvement in glycemic parameters, also help in enriching the beneficial gut microbiome and the depletion of the pathogenic microbes. However, this GSS supplementation can be used as an adjunct therapy for the individuals with T2D to restore the healthy gut microbiome. I thank my all the study participants and both the mentors, Professor Saroj Ghaskarbi from the Savitribai Phule Pune University and Professor Yogesh Sauche, for their support and guidance to the entire study. Thank you.
Thank you, Akshay. We now move to the last presentation for the third prize, and this will be by Aditi Goel from the Central Food Technological Research Institute, CFTRI. Aditi, please come. Thank you.
Good afternoon, everyone. So today I'm here to present my poster entitled Vitamin A Enriched Probiotic Carrot Nectar Regulates Gut Microbiota and Reduces Obesity, a leading cause of cardiovascular diseases. So obesity is a global health issue in the developed and in developing countries. And according to World Health Organization, presently there are 600 million obese adults, which almost represents 13% of the world population. And this number is expected to rise over 1 billion by year 2030. Obesity is considered as a root cause for many non-communicable diseases like cardiovascular diseases, and NAFLD, type 2 diabetes, and arthritis. So it is very important to detect and prevent the obesity at very early level using various markers like increased BMI and dyslipidemia. This will help the prevention of many metabolic disorders. Though FDA has approved many anti-obesity drugs, but they are associated with some side effects in the long term. And probiotics is widely studied for the anti-obesity effects. And combining these probiotics with the suitable food metrics can help in the prevention of obesity. According to a global healthcare company, a weight reduction of 5% will lead to prevention of hypertension and hyperglycemia. And if a person is able to manage the weight reduction of 10 to 15%, a positive effect can be observed in the cardiovascular diseases also. So to develop a natural anti-obesity therapy, we have screened several fermented food products collected from the Sikkim and isolated many probiotic bacteria. And finally, we have selected one with a good cholesterol reduction potential. This Lactobacillus plantarum MCC5231 was earlier characterized for the production of bacteriocin, which was active against a range of foodborne pathogens and multi-drug resistant bacteria. It is shown to have good sustainability in gastrointestinal tract based on its acidic and bile tolerance and binding ability to the mucin. And it is also characterized under in vitro conditions for the cholesterol reduction potential. And based on the European Food Safety Authority's guideline, if a probiotic bacteria is to be used for the consumption purpose, it has to be analyzed for the safety assessment, and after the following the guidelines of authorities, it is found to be safe for the consumption. So this is the probiotic carrot nectar, which is developed after the incorporation of probiotic bacteria into the carrot nectar. And this probiotic carrot nectar can be stored for 75 days under refrigerated conditions with a viability of more than 6 log CFU per ml. So to see the anti-obesity effect of this probiotic carrot nectar, C57BL6 mice were used, and they were fed with high-fat diet for first 8 weeks to make them obese, followed by the probiotic treatment for another 8 weeks. So the mice were divided into the 6 groups. Only first group was given the normal diet, rest all the groups were given high-fat diet. Once the mice gained appropriate weight, the probiotic treatment was started. In group 6, the probiotic product was fed, and in group 5, only probiotic bacteria was fed. Whereas in group 4, only carrot nectar without the incorporation of probiotic bacteria was fed to do a comparative study. Whereas in group 3, a reference bacteria, which is already studied for such kind of benefits, was used. Now, the measurement of physical parameters is first visual appearance, which tells about the effectiveness of the probiotic bacteria. So weekly body weight was taken during the during anti-obesity during the studies, and we found that the high-fi diet has increased the weight from 25 to 46, which is reduced to 82% on giving the treatment directly through the cells. Whereas this reduction in weight was even higher when the probiotic product, which was incorporated with the cells, was given. The similar effect can be seen in the reduction in organ weight, especially in the adipose tissue, where the weight reduction of weight was reduced to 77 to 75%. And similarly, abdominal circumference was reduced to almost 85%. Now, as we know that the gut microbiota is a major reason behind several such diseases, so to see the changes in the gut microbiota, the weekly fecal sample was collected and was analyzed for the changes in gut microbiota. So feeding the mice with high-fi diet resulted in gut dysbiosis, whereas when probiotic treatment was given, the lactic acid bacteria count was increased, whereas the harmful pathogens like Staphylococcus aureus and E. coli was reduced. Now, our aim was to count the number of Lactobacillus plantarum present, which can be a contributing factor for this. So we have done the quantitative PCR using Lactobacillus plantarum specific primers, and we found that high-fi diet was containing 4 log CFU per ml of Lactobacillus plantarum, whereas the probiotic treated group were containing 8 log CFU per ml of probiotic bacteria. Then the changes in serum lipid profile was measured. So the treatment with probiotic bacteria has reduced the total cholesterol levels to 81.3%, whereas in probiotic product fed group, the level was reduced to 75%. And similar changes can be observed in the levels of triglyceride and LDL, whereas the amount of HDL, which is considered as a good lipid, has increased. SGOT and SGPT are the markers of hepatic dysfunction. When there is necrosis in the liver, these markers are leaks out into the blood, which results in increased serum level of these markers. And this is what we have observed in our studies. In high-fi diet, the level has increased in comparison to normal diet, whereas on giving the probiotic treatment, the serum level of these markers were reduced. Now, as we know that the gut dysbiosis will lead to the oxidative stress and inflammatory conditions. So to see the immune response of the mice during high-fi diet, the inflammatory markers were checked. And what we have observed is during high-fi diet feeding, the inflammatory markers like IL-6, IL-12, and TNF-alpha were increased, whereas the amount of IL-10 was reduced, which is reversed on giving the probiotic treatment. And most of the probiotic bacteria have good application potential in reducing the oxidative stress by regulating NRF-ARE2 pathway. And so the giving the probiotic treatment to the obese mice has reduced the oxidative stress conditions in the liver. The oil red staining for the liver tissue was done to see the deposition of lipids. And in high-fi diet, the red color droplets represents the deposition of fat. As per the scoring, the 67% of the liver of high-fi diet group mice was accumulated with the lipids, whereas the probiotic treatment has reduced this accumulation to 37%. The similar findings were observed in H&E staining, where the second figure is the liver, and in HFD group, we can see the vacuolar structures, which represents the deposition of fats. The similar structures can be observed in NPP group, which was fed only with the carrot nectar without addition of probiotic bacteria. And these vacuolar structures were reduced in LP group, whereas when the treatment was given through probiotic product, these vacuolar structures were completely removed. Now to find out the mechanism how this probiotic bacteria will be working on the liver and adipose tissue, several obesity-associated markers were checked. And finally, we concluded that when there is gut dysbiosis, inflammation, and oxidative stress in the body, it will lead to the disbalance of leptin and adiponectin into the adipose tissue. And this disbalance of adipokines will result in disruption of metabolism in adipocytes, that is AMPK pathway, which was regulating the lipid homeostasis in the liver through PCSK9 and SREBP2. And therefore, now the liver is not able to metabolize the lipids properly, and it will be accumulating into the blood, leading to increased levels of fat in the body. And against this, the probiotic bacteria is working. So conclusion is probiotic carrot nectar is a symbiotic beverage where probiotic bacteria and fiber-rich carrot act symbiotically to regulate lipid metabolism and fat accumulation via modulation of gut microbiota and immunomodulation. Thank you.
So thank you so much, everyone. Now it's time to felicitate the young investigators. So I request our chairpersons to do the felicitation. So we'll start from Ayushi Purohit. I request Ayushi to come and join us on the stage. Ladies and gentlemen, a round of applause for Ayushi Purohit. Next, I request Manisurya Palepu. Next, I request Akshay Gaik. Next, I would like to welcome Aditi Goyal. So now it's time to felicitate our chairpersons. For that, I would like to welcome Professor N.K. Ganguly and Professor Chandrabas Narayana onto the dais. So thank you so much, Professor Jyoti Tamang and Dr. Sharad Gopalan for chairing the session Young Investigators presentation. So can I have the young investigators also to join us for the group photo? Also requesting Dr. Neerja Hajela, Dr. E. Sri Kumar, all Japanese delegates and all GB members of Gut Foundation to join for a group photo. So our top 20 young investigators can collect your participation certificates at the registration counter, and the delegates will get the e-certificates on their email ID. So after the group photo session, I request the panel members to stay back on the dais itself for the panel discussion. Thank you so much, everyone. So moving on to the panel discussion, I would like to welcome Dr. B. Sesiguran, former Director, National Institute of Nutrition, Hyderabad, India, to moderate the panel discussion.
So you can sit there, like that. Which side? One of the end you have to sit there. You want to sit this side? Sit there. That's it.
So as we begin our panel discussion, our moderator will be Dr. B. Sesiguran, former Director, National Institute of Nutrition, Hyderabad, India. And our panel members are Professor N.K. Ganguly, Professor A.K. Srivastava, Professor J.B. Prajapati, Professor A.P. Dash, Professor K.R. Lahiri, Professor Rama Chaudhary, and Dr. E. Srikumar.
So this is the grand finale of this symposium. Unfortunately, we have very little time left, so we'll have to speed it up a little bit, and I need the cooperation of all my panelists as well as all of you to make it short and sweet and as interesting as possible. I will now start from, I have a list of names here, as was mentioned now. I'll start from the bottom of the list and go up, because then Professor Ganguly will have the last word. Okay. So now Srikumar. Srikumar, of course, you know, is the co-organizer, co-host for this conference and is director of the Institute of Advanced Virology. Dr. Srikumar, I will be asking questions for each one of them, one question. And each one of you should respond to it in a maximum of 3 minutes. And there's a timer here. And subsequently, the audience will be requested to ask the questions appropriately to you. So, Dr. Srikumar, we have a current understanding. What's the current understanding of the role of the virome of the gut? We know so much about microbiome. Yes.
See, in fact, this virome is it's coming up. Maybe there were a lot of limitations in studying the virome. Unlike this bacterial species that we study, there are no signature sequences for identifying these viruses. So essentially, we need to identify the sequences and compare it with the database, so that we need to identify the viral segments that we pick up. So the NGS sequencing is getting refined. We are getting more and more longer sequences, so that, and also parallelly, the database is also getting built up. Maybe in the last couple of years, there has been significant index in the total virome database. Not only the gut virome, the metagenomic sequencing is coming up. So now we have something, these fragments that we are getting, we can compare with the database and identify the viral species in the gut microbiome itself. So that is going to be the first step. Now, second step is going to correlate these signature sequences with the disease conditions. So that is coming up slowly. So I would say that the virome is a new thing that is coming in the whole microbiome scene. And definitely, these are going to be super bugs. Like most of them are bacteriophages that will be regulating the bacterial component. So even when we say that, okay, one particular species of bacteria is going up and another is coming down, maybe there is another regulator that is controlling this. Maybe it's a bacteriophage that will be, most of these bacteriophages are lytic phages, which will be destroying these bacteria. And maybe some of them are temperate phages, which is into the lysogenic cycle. But definitely, this virome is going to be a master regulator sometimes, at least in certain conditions of disease, where this will be controlling the microbial population itself. So maybe the delay in building up the database and also the accuracy or the throughput of the genomic sequences that we are getting, that was one thing which was getting delayed. But now, more and more people are getting interested in the field and they see a lot of potential in that. And the COVID has done was a big trigger, especially the intestinal infection that caused by the virus was also a trigger why some people are getting the intestinal infection. Some are not getting only the respiratory form of the disease. So definitely this is going to be another, major addition to the microbiome research.
Thank you, sir. Thank you. Now I'd like to introduce Professor Rama Chaudhary, who was the former professor and head of the Department of Microbiology at AIIMS, New Delhi. Madam, you have a lot of experience relating to probiotics in the role in gastrointestinal infections, C. difficile diarrheas, and also inflammatory bowel disease. So can you share what your experience was?
So thank you for the question. I think it's a real vast question and difficult to cover in 3 minutes. But what I really want to say is there is a proven effect of probiotics as far as diarrheas are concerned, especially the rotaviral diarrhea or travel-associated diarrhea. But the problem has been in IBD cases. We did hear from Dr. Vineet about the FMT role in cases of IBD. But we also studied, probiotic strains, individual strains, and we have done 2 studies, which is a double-blind randomized control trial, and where we have checked the Bacillus clausii strain, and not only seen the microbiome in the healthy IBD patients and also some of the colon cancer patients, but we have also tried to see the inflammatory markers, because there was a question raised whether inflammatory markers were studied. So we individually studied IL-6, IL-10, and TNF-alpha, and we found that by giving this probiotic strain, the anti-inflammatory marker IL-10 was definitely increased. There were also, behavioral factors, that what patient feels, and that is the most important part of IBD. And clinically, we found that the patients felt much better. All the parameters, we had a questionnaire where they were answering before and after the treatment. And we found once the analysis was done that all these parameters were improved in cases of IBD patients where they were given probiotic. And not only that, we also studied a very important factor we have realized is that in cases of wherever, this probiotic was given, there was increased in some of the species of in the microbiome, and Faecalibacterium was one of that, in addition to Lactobacillus, whereas Bacteroides and some of the other strains we found Megasphaeria and others which were reduced. So it was a real vast study where we have already published it in Anaerobe Journal, and where we could show that, of course, FMT remains the good choice, but there are certain strains which are found to be effective, and one can, give these probiotics where FMT is not possible or not doable for whatever reason. Coming to C. diff, there has been a lot of controversies as far as Clostridium difficile is concerned, because once the disease occurs, the probiotic may not act optimally. So it is always prevention which is better. It is better to take probiotic along with antibiotics so that you prevent antibiotic-associated diarrhea rather than letting it happen and then think what to do. So that is the first goal. And I can tell you that one of my friend who never believed in probiotic or whatever I was doing actually told me that when she was taking probiotic along with antibiotic, she was feeling much better and she did not get this diarrhea. So this is our personal
Thank you.
experience. Thank you.
That's very interesting, ma'am. So now I will introduce Professor Kia Lahiri, who was the former professor and head of pediatrics at the KM Hospital, Mumbai. Madam, you know that there is a lot of influence of the maternal gut flora on the child. And what is the extent of the influence? Does it occur only immediately after birth, or it is for the life?
Mom matters and milk-oriented microbiota. I'll just be giving three major points, which are take-home messages for all our delegates who are present here, especially the younger generation. It's very important to note that developmental origins of health and disease is a society which is looking into the matter. Mother's horizontal gene transfer and the vertical gene transfers are very important because mother's second and third trimester of pregnancy sees a change in diversity and the microbiota. Now, what happens is the mother gains weight, there is constipation, there's gestational diabetes, and of course, there's preterm labor. That's a very important point because if the child is born preterm, you'll have the necrotizing enterocolitis and you'll have many other issues which will come up and lifestyle diseases later on. So how is it important? And in children, in the first 1,000 days of life, that is 2 to 3 years, the first initial infancy episode, you have the Enterobacter, then you have the Lactobacillus and the Bifidobacter coming on, the Clostridia and the other organisms which come in, and last is the Bacteroides. So it's very important to institute maybe in our already underprivileged mothers to give some dose of probiotic to boost her immunity, and a lot of latent research in Nature and Cell, which have been published in 22-23, have demonstrated, and this is a very good area for research, especially for the younger generations, and you could look into it because the lifestyle diseases start in utero, and unless the mother is healthy, the child will never be healthy. So it's very important. Main two is Lactobacillus and Lactobacillus uteri, LGG. These are the two important interventions which can be done at the early stage during the second and third trimester, and maybe early part of infancy to the child. A baby and an infant can be given at any age. These two safe, and especially Lactobacillus uteri and Bifidobacters, this can be instituted very safely. And I think a safe motherhood, hygiene, low stress levels, feeding the mother, the SDG society which is formed, giving her immunizations, and of course, breastfeeding, vaginal delivery, because we are into cesareans in a big way, which causes a lot of problems. Breastfeeding gives you the glycans, which was mentioned yesterday. And of course, no antibiotics. I repeat, no antibiotics, because babies do not need antibiotics in the infancy because they have all viral induced infections. So I think with that, if we look into these three parameters, we are safe enough to prevent a lot of conditions, because we cannot take of the vulnerable population in the mother and her malnutrition to date, because it will take a long time to come. Thank you so much.
Thank you. That was excellent. In fact, while we have public health program to improve maternal nutrition, we should also make sure it improves the mother's gut health also, because both seem to influence the child's health. Thank you, ma'am. That's wonderful. Now, I have pleasure in introducing Professor A.P. Dash, Vice Chancellor of the AIPH University, Bhubaneswar, Orissa, India. Sir, what do you think about the role of the microbiome on the vector-borne diseases, and are there any differences between the tribal and the rural populations?
Yes, thank you. Thank you very much. Basing on the geographical surroundings, there are three communities: urban, rural, and tribal. And you know, there are the tribal population in India exceeds 120 million people, and mainly concentrated in undivided Madhya Pradesh, Chhattisgarh, Jharkhand, Odisha, and northeastern states. And the tribal, their lifestyles are different. The lifestyle of tribal community, you know, the food, we know that their diet is fiber is rich in fiber, plant-based and fermented, while the urban, rural dietary methods are entirely different, but they're changing gradually. As some eminent person while presenting shown in slides, there is a rural migration to urban areas and industrialization. These affect the gut biome also. As coming to vector-borne diseases, it is difficult to give. There are many, the few vector-borne diseases very important in India, like malaria, lymphatic filariasis, visceral leishmaniasis, dengue, chikungunya, kala-azar, chikungunya, Zika, and Japanese encephalitis, etc. But coming to malaria, take an example. You know, when I was a MSc student, 71 to 73, I read a book, How Mosquitoes Find Their Host. It was a book, it was like a story book. But later on, when the researchers are found out, there are three things how mosquitoes find their host. Number one is color. They're attracted to dark color. If you sit in the garden in the evening with a person with a white shirt and you are a black shirt, you will see mosquitoes. Among you, they'll be flying. So then second thing is carbon dioxide content of the skin. And you know the third one? The skin microbiome of the human beings. They attract the mosquitoes. And then the microbiome or the mosquitoes they have, they determine the vectorial capacity of a species. I will give an example. In malaria, we all know malaria is transmitted by a 52 anopheline, by anopheline species, and there are 52 species in India. Only 10 transmit malaria. We give a theoretical explanation that those 42 species who do not transmit malaria, they do not live long to allow the parasite to grow. But is there a role of microbiome? We do not give any scientific explanation to determine which is a vector and which is not a non-vector. Again, if you see in tribal areas, Vivax malaria is less found than the falciparum malaria. Unlike rural and urban malarias, there also, I believe, something like microbiome might be playing a big role. And by exploring these things, we will know it will help us to reach the sustainable development goals. All of us know goal number 3 and goal number 2 and goal number 3. They belong to nutrition, good, sound health, and all these things. Thank you.
Perfect, sir. Thank you. Maybe someday we'll have mosquito repellents which have bacteria impregnated into them. So now I'll introduce Professor Prajapati, who was the chairman of the VCOE Institute of Rural Management, Anand, Gujarat, an expert in dairy and probiotics. Dr. Prajapati, are there any novel probiotic foods which are coming up in your field?
Yeah, thank you, Dr. Shishikar. Actually, when you talk probiotic foods in general and then something novel, probably some of you may be knowing that in the food sector, the fastest growing category of food is probiotics, nutraceutical, health foods. Because the society has learned a new word called as immunity after COVID, and everybody, the demand has increased. The overall global probiotic food market is growing at 8.1%. But in India, it is growing at the rate of 20.1%. That means it is going to be double in next 5 years. So all those who are processing foods and also pharma, they should be happy that they have a lot of potential. Now, when we call novel, novel is not only in terms of the food item which is launched. Novel is in terms of application, novel probiotic strains, which people are discovering at fast rate. Similarly, novel raw materials which are being used for making it, and novel applications which people are trying for that. And in all these things, one thing which has become super novel is application of genomics and then designer foods for that. So at the moment, we have almost 60% of the probiotic foods based on dairy. Because when you take dairy as a raw material, you have not to waste time in proving the nutritional value of that food because it's already established. But when you go for some other types of raw materials, you have to first say that, okay, this is a nutritional, this means fiber and this much iron and this. So that is convenient. Second thing is that milk protein gives very good buffering action, and that is why they survive for a longer time. So now, apart from milk and milk-derived products, we have also non-plant-based products also. We have products for frozen category like ice creams, also in chocolates, and also the products which may not have directly the probiotic in that, but there will be other material. For example, the probiotic straw. You have a juice, because in juice probiotic you will add, it will die. But the straw is impregnated with immobilized probiotic. So when you drink it, the required dose will go that. So novelties are going on. And the new strains which are being isolated, especially they are for specific application. And most of the strains, including Erkemenschia, is for immunity and so. But the other applications now, see, okay, probiotic considered as a remedy for all. So everybody is thinking probiotic like a magic drug, even though it is not at all a drug. And there are two sectors, food and pharma. As far as food products are concerned, fresh products are running, long life products are also going, and there are application of bacillus spore formers also in that for heat treated products also. But in pharma products, there are a lot many spurious products, and I want the physicians to be careful in recommending such products without knowing that what is clinically proven for those products.
Thank you.
so
Thank you, Prajapati. Now, we now go to Professor A.K. Srivastava, whom you can see on the screen. He is online and is Vice Chancellor of Duvasu in Mathura, UP. And you know, Mathura is the home of Lord Krishna, and Lord Krishna is associated with dairy products. And now we'll ask, are dairy products the best food matrix for probiotic delivery? If yes, then you have to tell us why.
Thank you very much, Dr. Sashikaran. Thank you very much. At the outset, I apologize that because of my presence here, I could not attend physically, although I was having very strong desire to be with you physically. Thank you. As far as your question is concerned, I tell you that long before the existence of pathogenic microorganisms, the ability of beneficial bacteria to convert milk into long-lasting fermented products and its health attributes were recorded, and that to 6,000 years back. As far as your specific question is concerned, I can say that yes, in this scientific era, we are having a lot of validation, lot of data to say that fermented dairy product is the best food matrix for administration of probiotics in human being. I will not take much time. I will give just 6 points to prove that yes, it is the best food matrix. Number one, means there are some important genes which are encoding the probiotic functions, and these are induced only during the process of fermentation. Otherwise, they are not induced. We will not get their nutritive therapeutic effects if they are not going in the process of fermentation. So there are important, at least 5 or 6 genes have been already identified, and people are working on more genes which are expressed only during the process of fermentation. Second point, as we know that many organisms, pathogenic organisms, or many means probiotic organisms, they do not survive in acidic gastric pH. On one hand, on other hand, the fermented dairy products, they are having the inherent properties to neutralize the gastric acidic pH. So it increases the survival of the probiotic bacteria in our GIT. And third, we know that we are always going for refrigeration of our fermented dairy products. So it will be an addition for survivability of the probiotic bacteria. Third one, we all know that there is a positive perception of the consumers about the dairy products, and they know that there are healthy live bacteria. Consumers will always prefer to take probiotic bacteria as a food or in food than as a medicine in form of the sachets and capsule. And fourth, which is more important, and I have a lot of information, lot of recent publication, and very recently it has come in a very big way from the New York State University, Buffalo, and they said that lactoferrin, which are a nutrient molecule in the milk, lactulose, many bioactive peptides, galacto-oligosaccharides, they are having synergistic effect for the pharmacological effect of or therapeutic effect of probiotics. Synergistic means 1 + 1 is equal to 3. Galacto-oligosaccharides, lactulose is also acting a very good prebiotics, which enhances the multiplication of probiotic bacteria in our intestine. Fifth one, although we can debate that yes,
another minute.
1 minute. Although we can debate that S. pylori is good or bad for the intestine, but when we talk about the eradication of the S. pylori, then we found that probiotic in dairy products is several fold better than it is given in the sachets or tablets. And last point, recently we know that the first probiotic, clinical probiotics which are available in the market is present in the fermented dairy products. That is the Cardio Viva. It is for the cardiovascular health. And then in pipeline, we are having the second probiotics, clinically proven probiotics, and that is also in fermented dairy products, and that is the Glucoviva, which can take care of the type 2 diabetes. So in nutshell, yes, it is the best food matrix for administration of probiotics. Thank you very much, sir. Thank you.
Thank you, sir. I think after Varghese Kurian, you will be the next milkman of India.
Thank you. Thank you.
The last, but definitely not the least, and Professor N.K. Ganguly, I don't think I'll waste my energies introducing him. Sir, this is one big issue which is, I do not know how much of it is just hype and how much is truth, is that non-nutritive sweeteners, they say are not good for the gut bacteria. What do you think is the safety and what's the impact of these on the gut?
Since I have the mic, I will thank my brother Chandras and Sri and the whole RGCB family for this fantastic experience which you gave us, and all the youngsters and the scientists of the Trivandrum who have sat through the two days of our deliberations. Thank you very much. Then next, I will like to thank all the scientists who participated in the poster study. It was very difficult. I don't know who became first, who became second, who became third. Actually, all of you were winner. It was very difficult for me when I went through. At least I could have made another 5 winner. We will write a letter of appreciation individually to you that your work really made an impact. So with this now I come to answer your question. There are about 20 sugar sweeteners. Out of that, there are 6 or 7 which are most. They go with different names, but like aspartame family goes by name of equal. Then we have sucralose, we have saccharin. Saccharin was the first one, and then we have the stevia glycosides, and we have other fruit ones, monkey fruit and other fruit, which are, and of course the glycyrrhizagabra, which we call glycyrrhizine, which is known as, in India, known as moletti. So all of these are non-sugar sweeteners. I was helped by Rizwana Hassan and Ayushi to scan any paper which has been published in this particular area. And it was so confusing. So some said that it does not have a bad effect. Some said it has a bad effect. There were meta-genomic analysis also. So I will say that all non-sweeteners, non-sugar sweeteners are FDA approved, all of them. The saccharin was initially withdrawn, but whole Europe takes saccharin. It was initially withdrawn with association of bladder cancer. That particular thing has been removed now. Aspartame in 2023, there was a committee which was set up, and this has also cleared the aspartame. So as far as official position is concerned, all are safe. However, now I can go to my analysis. However, there is an issue that these non-sugar sweeteners, many of them have added zinc sulfate to this, because it leaves a aftertaste. This aftertaste is bad. And those who know what zinc sulfate does, it is used for halitosis. It's a very nice thing. So when you add zinc sulfate, many of the bad effects of these go away. In this, many of them are non-metabolizable. So those who have been fermenting microorganisms, as I have been doing in the industry, they need an energy source that is provided by the glucose, metabolizable sugars. So all of these non-sugar sweeteners, they don't provide that. Only, but the safest one is the stevia glycosides, and Yakult adds a little sugar into that. And that particular sugar is both for aftertaste as well as these microbiomes which are given to survive. Rest of the others on the aspartame group actually is an excitome also. It in a large amount, it's at the same group as monosodium glutamate, Ajinomoto, and it causes flushing to someone, some people, and it is an excitome which actually may excite NMDA receptor, cause calcium flux, and cause the brain damage, but it is only in the infant if you give in a huge amount. So in larger amount, it doesn't do. The bad ones are sucralose and saccharin and others. They have bacteriostatic effect on the microbial population, and bacteriostatic effect is much worse than the bactericidal effect. The second effect which they have, that they also damage the epithelial cells. And epithelial cell, we were talking of the crypt of Liverpool, all those gastroenterologists and pathologists who are sitting, they actually replenish the epithelial cells. So these also act on the crypt. And they also act on the tight junction, increase the permeability, affect the nutrition, and they're not metabolized. However, there could be a cure for that. In the notobiotic animals, there are very good studies where the responders and non-responders, if you put those to other mice and give them a very heavy dose of non-sweetener sugars, the responders behave well and non-responders do not behave well. But there are a lot of individual preferences. I eat all the bad things in my life and I take sweetener, but I will advise no healthy person to take sweetener. Because healthy persons, my wife takes it because she wants to. WHO guideline is there. Don't take it to reduce obesity. Don't take it to reduce calories. But if you take non-sweetener rabdi, the most calories will be added by rabdi or kalakand or whatever. They're all over there. So these are the two dictums which WHO new guidelines have told. So many of particularly the ladies who are healthy and young, they take it, and my wife also takes it, and I'm going to tell her that don't take it anymore because it causes fasting glucose rise. It causes the glycemic index, after the food. The glycemic index should come down 40%. But here, the glycemic index is affected. So metabolomes are affected by the non-sweetener group. And one of the other thing which it does, that it causes some kind of a problem in the gastrointestinal neurons, which I was talking about yesterday. It causes a bloating sensation. It causes a sensation of fullness. So if you really want to, if you are a diabetic and you want to have a taste of sugar in your mouth, you can take it because you are taking other antibiotics. But in the other anti-diabetic drugs, few of them which are known to be good, like metformin is considered to be good. The SGL2 modulators considered to be good. There are others who can also promote cancer, PPAR1s and others. In the arena of the diabetics, you could balance it by taking it, and you may not be harmed. But essentially, if you can, because I have talked for many non-sugar companies, but since I was talking bad, they don't call me anymore. But the problem, my advice will be that avoid non-sugar sweeteners. Have black coffee, have tea without non-sweetener. But if you are healthy, don't take it.
Thank you, sir. Thank you. And as I mentioned right in the beginning, Professor Ganguly's word will be the last word. So we'll conclude this session right here. And then if any of the audience have any questions, you can address it during the lunch break when you meet our panelists. Thank you. And it was indeed a pleasure to conduct this panel. Thank you, all of you.
Thank you very much. Thank you, Dr. Sashikaran.
So thank you so much, everyone. So we have the felicitation. So request Dr. B. Sasikaran to felicitate all our panel members. Yes. So the felicitation for Professor N.K. Ganguly. Ladies and gentlemen, a round of applause. Next is for Professor A.K. Srivastava.
I received from here. Thank you. Thank you very much.
Thank you so much, sir. So next is for Professor J.B. Prajapati. Next, Professor A.P. Dash. Next, I would like to welcome Professor K.R. Lahiri. Next, I would like to welcome Professor Rama Chaudhary. Next is Dr. E. Srikumar. And I would like to welcome Dr. Jagmeet Madam onto the dais to felicitate Dr. B. Sasikaran. So request everyone to gather for a group photo.
Please add my photo also in inset.
So thank you so much, everyone. Ladies and gentlemen, a round of applause for that panel discussion. So moving on to the vote of thanks, I would like to welcome Dr. K.B. Harikumar onto the dais.
Good afternoon, all. So I am the person standing between all of you and the lunch. So I will finish my duty at the first. So I have given one vote of thanks, but this is important. So I'm happy that the 2 days scientific deliberation finally come to an end, and I hope all of you have a wonderful time here and gain more knowledge about the current developments in probiotic research. So first, on behalf of the organizing committee, I express my sincere gratitude to all the speakers, all the foundation members of the Probiotic Society, and all the colleagues from Japan, all the young investigators, all the delegates for their time and attending the meeting. Once again, my sincere gratitude to Professor N.K. Ganguly for his support and constant encouragement. I also take this opportunity to thank the Elbourne conferences, the event management team, Mr. Rajiv and Mr. Karan, for their timely help and for making all the necessary arrangements. And to make this entire event successful, there are several people from RGCBs involved. It's not a good idea if I'm not mention their names. So I express my sincere gratitude to Mr. S. Mohanan Nair, our Chief Controller, Mr. N. Jayagishnan, the Chief Manager, our Finance Officer, Mr. Sadi Chandran, and our Chief Deputy Controller of Finance, Mr. Kumar, for all their help. So you have seen all the entire audio visual arrangement. So this was entirely possible because of our instrumentation team from RGCB, headed by the instrumentation head Rajshekharan and his entire team. And I also would like to thank all the student coordinators from RGCB, the PhD students and research fellows. Basically, they were actually helping in all different ways, inside the hall and outside. So I cannot take all their names. It's a long list, but I personally thank all of them for standing with us all throughout the 2 days. Apart from this, I also would like to thank the RGCB canteen who are actually serving you this the food items, and the administration and the security division. And finally, I again extend our sincere gratitude to our director, my director, Dr. Chandrabas Narayana, for giving me an opportunity to participate in this and all his support. And I also like to thank Dr. Sri Kumar, the co-organizer, and Dr. Sabu Thomas, who is from the Center for Excellence in Microbiome. Thanks again for joining us. I hope you all enjoyed the meeting. Thank you.
Thank you so much, Dr. KB Harikumar. So as we wind up, so as the scientific symposium concludes, I extend my gratitude for your active participation and insightful contributions. So let's carry this momentum forward and continue advancing knowledge in our respective fields. So I request everyone to stand up for a minute for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उज्जल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल नायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे।
So, ladies and gentlemen, for your attention, the lunch for speakers, foundation members, and special invitees is at the CV Raman Hall besides the poster display, and all others can have your lunch at the Pantal. So, our top 20 young investigators, you should ensure to collect your certificates from the registration desk, and all the delegates will receive your certificates through your email ID. So make sure that you all are submitting your survey forms without fail at the registration counter. Thank you so much, everyone, for joining.